And my sound is way low. Let's turn that up. All righty. Good afternoon. Today is Thursday, April 15th, 2021. Technically a national holiday, but not normally this year because, Corona. I got a little worried when I was looking. There's only 16 of us here right now. I thought maybe the last exam scared a lot of people off, but no, we still are enrolled at 25. So people are maybe either sleeping in or something else. Maybe for this last section, we'll have a few more daily quizzes just to make sure people are here and on time. And maybe we'll make them worth a little bit more points too. So we'll see how that goes. But good news. We are on to our very last section of this class. So of the 5 sections, we are now on the 5th. Bad news is it's a lot. We are trying to cram the nervous system into basically 2 months of information, 7 lectures, I think, or something like that, month and a half that we have left in this class. And it's a lot of material to cover. And in some ways, it's not as intuitive. Right? When you look at the shape of a bone, you can sometimes get an understanding of its function. When you look at the shape of a muscle and the location of the muscle, we absolutely positively could use that to figure out what it does. However, the part of the brain that moves your hand, does it look like a hand? Or the part of the brain that you see with, does it look like a big eyeball? No, of course not. So when we look at the nooks and crannies of our nervous tissue, it isn't necessarily going to intuitively tell us what it does. There's also something very, very important about the nervous system that we have to understand right from the bat, and we're going to continue to cover through. We will get as far into the special senses as we can. We'll see where we are. This section tends to run late, so absolutely we're covering the central nervous system, nervous tissue, the autonomic nervous system, which I know is near the end of the class, but it's probably the stuff that's going to be the most important when you move on to 431. And we'll get to the specialized senses if we do. We'll get as far into them as we can based on where we are in the class at that point. So we'll see how far we get. But one of the important concepts to remember about the nervous tissue is one of the things that we have said again and again and again in this class is that structure and function go hand in hand. Right? The shape of something affects how it functions. The function of something affects how it looks. And again, probably the classic example we see of that is in our muscle. Right, the shape of that fan-shaped pectoralis muscle, pectoralis major, shows us the wide range of motions it has, whereas that brachialis, much more linear, just has the one action. The other thing is, right, this muscle cell here in my arm and this muscle cell here in my leg essentially do the exact same thing. Yes, they move different parts of my body, but they contract and they produce tension, and they essentially do the same things. Does this part of my brain and this part of my brain necessarily do the same thing? No, and in fact, this part of my brain and this part of my brain don't necessarily do the same thing as well. So we have both lateralization, one side can do something different from the other side, and specialization. Here may be where I move my leg, and here may be where I touch with my hand. So there is this relationship between structure, function, and location. We are going to add a third layer to that. In fact, if you look at your homework assignments here, and this is not all of them, this is just everything we've got going on in the month of April. But at the very end of April is this nervous system review, which I purposely have not made available for you yet. I'm sure everybody here has already gotten onto Canvas and looked at all of the materials that you are responsible for this section. And when you did that, one of the things that you noticed is that this handout is not available yet. The whole point of this handout is to really emphasize this relationship between structure, function, and location. So the nice thing about this is if we know a location of a neuron, where that cell is located, based on its location, we're going to know what it does, and we're going to know what it looks like. And so conversely, if we know what a neuron looks like, we should know what its function is and have an idea of where we would find it. These three pieces of information are going to get together, and this handout is going to help you to do that. It is indeed graded for correctness, but the problem is we haven't talked structures yet, we haven't talked functions yet, and we haven't talked locations yet. So clearly this is not something I expect you to be able to do right away, which is why it's not due to the end of this month. So we have a couple weeks to start learning the structures and the functions and locations, and then it'll be much, much easier to put those pieces together. Let's see, do we have any tips for us as far as studying goes for this module? I'm not sure what the crash course is. What is crash course?
It's like a YouTube channel that like he goes over like information basically on different things.
Oh, okay. Again, I'm all for using all the different modalities you possibly can. However, this is a massively large section, right? Again, as I mentioned, my PhD is in neuroscience. I spent 7 years of my life studying the nervous system. So clearly, we're not going to be able to cover everything in this about the nervous system in 7 lectures. So really, the key to this is well, there you go. That means my lectures are a complete waste of time. If I just made them 10 minutes long, that would make my life a lot easier. I wonder if I get paid the same if I did that. I'll have to look into that. But obviously, focus on the things we talk about. The things we talk about are the things that we are going to be emphasizing in this class. And I could see where it would be helpful for something like the skeletal system, because bones are the bones of the bones of the bones. Muscles are the muscles of the muscles are muscles. Nervous system, there's a lot more information we're going to try to cram in here. So really focus on the things we talk about in lecture. All right. Excellent. So as you can see, we have a fair amount of stuff to talk about today. On Tuesday, again, we do have lecture, even though it's a holiday, and you have 2 assignments that are due, including a Labster. Thursday, you have Unit 13 review, and I know how much you guys all love that Physio X. If there's one thing students tell me time and time again, is they absolutely positively love the Physio X. So absolutely, you will get that opportunity to work on that. And then this next activity has 9 different activities, which means 9 different lab reports. So again, thankfully, you can just save them as PDFs and submit them to the website, so you don't have to kill a small tree cutting them down. Yes, there was a question?
Yeah, so you assign Physio X exercises, and yes, we all hate them, but I find the Labster simulations a lot more helpful. Is there a reason why you choose Physio X over Labster?
Well, if you notice, I don't pick one over the other. I use both. So you
Yeah,
have
but
activities in both.
I think we've only had like 2 or 3 Labsters so far.
Well, and clearly you've only had 3 Physio X's. Again, what I will say is there is not a huge difference between 430 and 431. It's basically a continuation of both classes. However, if you think about the very beginning of this class was tissues, was bones, was cells, was all of those types of things that there's not a lot of physiology necessarily associated with them. When we get to the next organ system, when we get to next class, 431, when we're talking about respiratory system and digestive system and endocrine system, there's going to be a lot more physiology. So there'll be even more Labsters and more Physio X's that you will be doing in 431. All right, well then, do the Physio X's first. All right. Well, see, there you go. A little bit of something for everyone. That's the advantage of having this. Everything's not necessarily going to help everybody, but the more resources I give you, the more likely you'll learn this material. All righty, excellent. So that is the game plan of what we have going forward. Let's dive into our lecture. As I mentioned, we were talking about the nervous system. And if you remember all the way back at the very, very beginning of this class, we learned that there are 4 major types of tissue in the body. What were those again?
Muscular, nervous, cardiac.
No,
no?
cardiac falls under muscle.
Lymphatic.
No, tissue types, not organ systems.
Bone tissue?
Say again?
Bone tissue?
Now, bone falls is a specific type of what general classification of tissue?
Yeah, I don't remember the other two.
Anyone help her out?
Active.
Connective, there you go. And what's the fourth?
Nervous.
Well, so someone said nervous, we've said muscle, we've said connective, so what is left?
Blood.
Blood, remember, is a connective
Body
tissue.
These.
This is a cumulative class, right? I
Fat
thought this
tissue?
would be an easy question.
Is it fat tissue?
Epithelial.
Epithelial, there you go. Epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Exactly. I thought that would have been one of the easier questions. Remember, our whole second test was practically on epithelial tissues and connective tissues, right? We saved our discussion of muscle tissue till we got to muscles, and now we'll do the same thing with our nervous tissue. We will talk about nervous tissue now that we're talking about our nervous system. What is the function of our nervous system? What is the function of nervous tissue?
Communication throughout the body.
Absolutely, right, exactly. It, along with our endocrine system, remember, are the two systems that are specialized for communication. They allow us to be able to receive information, respond to our environment, and these two basically are what we use to maintain our health and maintain our homeostasis. And in particular, how is it that the nervous system communicates?
Through electrical impulses.
Exactly, it produces large, positive electrical signals. And if I remember correctly, we have a fancy name for that big positive electrical signal. What was it again?
Action potential.
There we go, action potential. Spectacular, we have that action potential. And absolutely, if we wanted to be even more precise, because we started with them in muscles, neural action potential, absolutely. It's one of the reasons I like doing the nervous system first. With the nervous system, we get a little introduction to a baby action potential. And then when we get to the nervous system, we get to talk about the big boy and talk about it in great detail and length. Excellent. However, while that is a vitally important function of our nervous tissue and our nervous system, it does a tremendous amount of other stuff for us as well. It allows us to perceive our environment. It is responsible for our emotional state of mind. It, as we learned in the last section, stimulates the movements of our muscles, provides us with our memory, and so much more that it does for us. Our nervous system is a truly, truly remarkable organ system, absolutely positively. And of course, the study of the nervous system is the field of neurology or the field of neuroscience. All right, so our nervous system, when we think of it from a functional standpoint, and that's, let's do it this way. We'll start here. So we are going to talk about the organization of the nervous system. As we have already kind of hinted at, we are going to be able to divide that basically into, and I think, whoops, hold on, that was a little too high. Bring that down. That should, oh, don't have to be red though. That might give us enough room, maybe a little bit more. We can talk about the structural organization. And we can talk about the functional organization. So here's our structural organization here. And then over on this side, we will do the functional organization. Excellent. All right. For our functional organization, there are basically three main branches to our functional organization. The first of these, and let's do function in red. The first of these is a sensory function. Sensory is basically where we receive information. From where?
Brain?
Well, with our sensory, are we necessarily receiving it for the brain, or is the brain where we're going to send it?
Or, I'm sorry, or
We receive it from like the whole body. wouldn't
Okay,
we?
absolutely. From inside of the body. Anywhere else?
World.
Yeah, and from outside, from inside and outside. Absolutely. Right, we receive information from the inside and the outside of our body, and that is absolutely positively our sensory function. All right. Now, as someone pointed out, we then take that information and we send that information into basically our integration center. Right? If you think about it, when we were talking about homeostasis, this is the part we referred to as the command center. And if you think about it, with this integration center, basically its job is threefold. First, it has got to receive information from either inside or outside the body. Then it has to process that information. Part of that processing is comparing it to what we know, to our memory. And then we have to make a decision based on the information. Right? This is our function of our integration center. So we've got our sensory, we're receiving information from the outside world. We have our integration center that receives the sensory information. I guess we should have said that. Here, I'll cheat. Once it receives that sensory info, it makes a decision based on that info, compares that information to what we already know, processing that information and make a decision. Then once it makes a decision, it that sends that information, or sends, information. We could say that because it is specialized for sending information. And where it sends our information to is to our motor function. And that motor function, its job is to, again, receive that information from the brain and affect some change. Right? Typically to maintain homeostasis. Again, this isn't a new concept. We've talked about this before, right? We know how this works. You're sitting there in your chair and you perceive that a bear with an axe walks into the room. You send that information to your integration center, which processes that information. Bear, bad. Axe, bad. Bear with axe, very bad, right? And so then we make a decision based on that. That decision is usually twofold. I either have to fight that bear with an axe or I have to outrun somebody in the room with me. Right? And so this are the three main branches of our functional organization for the nervous system. All right. Questions on that?
I have a question. When people get severe brain damage, do they lose the perception of their senses? Like they can't tell if they're hungry or the temperature of the room, or that's not how it's affected?
Because of the specialization of the brain, where the damage occurs is going to determine what the damage is going to be, what the deficiency is going to be, right? As I talked about, it's two simple areas. These two simple areas, one controls my leg, one controls my arm. So if I have damage in the brain where the leg is, then my leg's not going to work anymore. If I have damage where the arm is, and the arm's not going to work anymore. So again, it's all about location. It's not just structure and function, but location. The part of the brain that is damaged is going to determine the deficiency. And in fact, One of the things when we start talking about the brain areas and what they do, I think that damage or deficiencies in those areas is one of the great ways to understand and appreciate what it is that we do. So that's actually something we're going to talk about a lot. Laura, you had a question as well?
Yeah, so like in terms like say so like kiddos that like are artistic and have those type of developmental delays, is their sensory input or motor output delayed as well, or is that not have that doesn't have anything to do with it? Like because they don't know if something's good or bad, you know what I mean?
I do understand what you're saying. The short answer to that is it is far more complicated than that, but typically those types of cognitive deficiencies are occurring at the level of our command center, our integration center. Now, that doesn't mean that sensory structures coming in can't be damaged. For instance, individuals with Down syndrome typically have damage to their retina as well. So their eyesight tends to be relatively poor. So obviously, if you don't have good eyesight, that's going to affect your ability to receive information and bring that information in. And then your decision making, your processing, and all of that can obviously affect what you're influencing from the effectors going out as well. So it's not a simple answer, but the simple answer to it is that most of those kind of cognitive deficiencies occur here. That doesn't mean there aren't problems out here in the sensory and the motor, but most of the major cognitive malfunctions we think of, or deficiencies we think of, occur in this integration center. All right. So, excellent. For newborns, most of this isn't fully developed yet. The nervous system typically isn't fully developed at birth. Vision, for instance, is very, very poor at birth. They're obviously their motor function, right? They can't even hold themselves up, let alone, talk, let alone feed themselves or things along those lines. So all of this is something that develops as we age. Absolutely. Humans are born relatively helpless when you consider, for instance, like a deer or a horse. If you've ever been at a barn where a horse has been born, that newborn colt is standing and walking around typically within the first hour, usually much earlier than that, right? I'm yet to see a baby stand up and walk around after the first hour after birth for a human. So yes, many of our cognitive functions are not fully developed in that newborn. Most, I would argue. All right. And you guys are hitting on some good points, but we need to dig deeper into this material, the sensory motor and integration center, because let's start easy with the motor, because I think that's something we should already know. With our motor, as we've talked about before, basically that information that comes out from the integration center, the info goes to effectors. And what are the types of things in the body that we have said can be effectors?
Gland.
Glands, excellent. So one of them is glands. What else?
Muscles.
What kind of muscles?
All of them.
Like your heart.
Skeletal.
Okay, so I heard skeletal. I heard heart, so that would be cardiac. And any others?
Oh, and
Organ?
smooth.
And smooth,
all of them.
absolutely. Excellent. So basically all three of our muscle types and glands are capable of being effectors. So let's do that and that. Excellent. All right. Perfect. Let's talk a little bit more about our sensory as well. We just mentioned, for instance, that we can get information from inside and outside of the body. When we're dealing with info from inside of the body, right, this isn't always necessarily information that we are consciously aware of. Right. For instance, can you tell me right now exactly how much calcium you have in your blood?
No.
What the pH of your blood right now?
No.
The temperature even of your blood right now?
No.
No. However, do you think that those are characteristics that your body is paying attention to and monitoring and modifying to maintain?
Yes.
Yeah, absolutely. So to do that, we need a special type of receptor, and that special type of receptor is what we call an enteroreceptor. That oops, no, I did that wrong. That enteroreceptor basically is telling us information about what is going on the inside of our body. Whether it's body position, where our arm is, how stretched out our arm is, pH of the blood, temperature, all of those types of things. So there are special enteroreceptors that allow us to do that. However, we also have, as we've talked about before, our somatic sensory receptors. These are the ones that we find a lot of in their skin that give us general information about the body. Pain, temperature, right? And again, I know pain and temperature aren't things that seem like they're general information, but when someone has a heart attack, where do they usually grab? Do they usually grab their heart?
Their arm?
Yeah.
Yeah, they grab their left arm. They feel the pain radiating out here on the left arm. When you've had that 14th chili cheese dog, right? Do you reach down here to your stomach and grab your stomach in pain? Or do you feel that heartburn up here a little bit more, right? Things along those lines. So our general sense and temperature, we already talked about temperature when we were talking about the skin, right? If I put one hand in hot water and one hand in cold water, and I put them both in room temperature water, do they both feel the same? Does the water feel the same to both hands?
No.
No,
No.
those are general somatic sensory information. This is general information about the body, right? And notice I can still, even though temperature is a generic sensation, I can tell it with my elbow, I can tell it with my finger, I can tell it with the top of my head, I can tell it with my big toe. All of those areas aren't necessarily all equally sensitive, but they can all feel temperature. Can you hear with your big toe, with your elbow, with the top of your head?
No.
No. So not only do we have somatic receptors, but we also have special sensory receptors. These are specialized structures for a specific type of information in one or just a couple areas. So when we think of sensory, if you think about it, they're really three subdivisions to our sensory information. Internal information, somatic information, and special sensory. Can someone give me an example of a special sensory receptor where we might find something like that?
Vision.
Vision, absolutely. Vision is a great example. I already gave you the example of hearing, right? Hearing we just do in two locations. Smelling, taste, right? Yes, question.
Yeah, sorry. Could you remind me what somatic means?
Body. Soma refers to body. So this is things like temperature, pain, tickle, itch. Those are kind of more general sensations about the body.
Okay, thanks.
Yeah, because again, one of the big differences between a somatic sense and a sensory and special sensory is it in more than one location? I can feel temperature basically from the top of my head to the tip of my toes. I can only see with one organ. Well, two, the eyes. And again, thought experiment only. If you actually do this, I will take participation points away from you. So I want you doing this only as a thought experiment. So you wake up first thing in the morning and you get a little aggressive with the rubbing of your eyes. You didn't sleep well last night, so you get really aggressive with that rubbing of your eye in the morning. And what happens if you rub on your eye too hard? Don't do it, right? But if you were to theoretically rub your eye too hard, what would happen?
You see like white spots or
Yeah,
like
you see light. There is no light there. Yes, I guess if you do it hard enough, you cry. Yes. Notice there's no light. You could do that in the complete dark and don't, but you could do that in the complete dark. And even though you're putting pressure on the eye, what your brain thinks is happening is that you're seeing light. Because when you put pressure on the eyeball, that puts pressure on the retina and it stimulates the retina. And the retina is a specialized structure whose job is to see light. So when you put pressure on it, it fires action potentials and sends those action potentials to the brain, and the brain thinks you've seen a light. Right? Now, putting pressure on the retina can damage the retina, and those cells don't repair themselves, so don't do it. But it shows you an example of how these are highly specialized sensory structures for just a very specific function. And so that's like smell, taste, hearing, right, and seeing. Laura, yes, go ahead.
Yeah, so I'm not sure if this is a different part of like the nervous system. So for migraines, like when I get migraines, I also see like little light spots. Does that have to do with like the nerves or the cranial nerves, or is that something completely different?
You are right. Phantom lights are a very common symptom associated with migraines. The problem is the causes for many migraines are not fully understood. And since we don't understand what causes them, we don't necessarily understand why those symptoms occur. It may have to do with either an increase in intracranial pressure. It may have to do with differences in blood flow that could cause the cells to become active. But as far as I'm aware, there isn't a full understanding of why those symptoms occur.
Okay.
Yeah. And the problem is there are many different types of migraines. And causes. So I think it varies dramatically from person to person. Some they can be, an hour. Some can have migraines that last for days. Yes, Yulia.
Yeah, I was going to ask, so when people lose like one of the dominant senses, like vision or hearing, they tend to develop very sensitive sensors in the other area. Is that something that we do like consciously by choice, or is that just the way our body adapts to the new challenges? Like,
No, you're
let's say,
absolutely right. All you have to do is lose a symptom, lose one of your senses, and suddenly you can become a superhero. Right? You can hear through walls or see through, people's clothes or whatever it is. No, that is a complete inaccuracy. Many studies have looked at this when people who have either been born deaf or born blind, for instance, or become blind from some type of accident or something like that, their hearing doesn't get better. They're not better attuned. They don't have a higher sensitivity or anything like that. The only thing that is different is they pay more attention to it, right? We are very dominantly visual organisms. So we tend to pay more attention to what we see and less to everything else. So when you lose a sensation like that, you are more reliant on the others. So you have to pay more attention to them, but your senses don't get better. All right. Excellent. Yep. One of those great fallacies you hear about all the time. No, that people's senses don't get better. They just get more attuned to using them. All right. So now, of course, we know that there are 3 subdivisions to sensory. And guess what? There are subdivisions as motor as well. And those subdivisions of motor, A, we're already aware of, and B, are based on the effector they control. Since we just literally finished doing this, someone remind me what the branch of the nervous system that controls our skeletal muscle is?
Central nervous system?
Well, no, but remember the central nervous system produced an action potential and sent it down a neuron to the skeletal muscle. What type of neuron did we say it was? Does anybody remember?
It was autonomic, or the opposite of autonomic.
The opposite one of autonomic. It's the motor that has to do with the body. So based on what we just learned in sensory, what was it again?
was it somatic nervous system?
Somatic motor, absolutely. It was our somatic motor nervous system, which controls our skeletal muscle, and it controls something else about our skeletal muscle. Somatic motor controls contralateral, right? And then all the other cardiac muscle, smooth muscles, and glands, as was pointed out, is controlled by the autonomic nervous system. So our somatic motor and our autonomic nervous system. And I think we did talk about this. I'm not sure if I remember or not. Not only is autonomic a subdivision of motor, but it has two subdivisions of its own. And what are those two subdivisions of its own? Anyone know the two branches of the autonomic nervous system?
Is it sympathetic?
There you go, sympathetic.
And parasympathetic.
And parasympathetic. We will talk about these in great, great depth in the last couple lectures of the class, but there are some cutesy little mnemonics that people often use, some little rhyming mnemonics when talking about sympathetic and parasympathetic. Anybody remember what they are? What's the cutesy little mnemonic for the sympathetic? Helps us to deal with that stress when that bear with an axe walks in the room.
Fight
There
or
we
flight.
go. Ariel's got it, fight or flight. And what's the cutesy rhyme for parasympathetic?
I remember you saying it, but I forgot.
There we go, rest and digest. There you go, rest and digest, excellent, absolutely. So there you go. Notice here we have our three major functional divisions and all of the subdivisions that go along with it as well. All right. Questions on that? All right, then let's do something similar for our structural organization. What are some of the major structures? Let's talk about that first. Oops, let's change colors. Let's do blue for this. What are the major structures of the nervous system?
Spinal cord.
Spinal cord, excellent. I'm surprised that was the first one. I figured, yeah, usually brain is the one that people come up with first. Absolutely. The first two major structures, they're usually the first two that anybody comes up with and identifies, are indeed the brain and the spinal cord. Excellent, excellent, excellent. But they are not the only structures. Anybody have some idea of what some of the other structures of the nervous system might be?
Is ganglia one?
Absolutely. Ganglia, plural, ganglion, singular. What is a ganglion? Do you have any idea? Anybody know the definition of a ganglion? Well, here, let's do this. Let's move all this down a little bit. If you remember correctly, when we were talking about our neuron, and again, we'll talk about this in much more depth as we move our way forward. But if you remember, we talked about a neuron very briefly when we were in the nervous system. Basically has a large cell body. And let's make this small. It has a large cell body with a bunch of processes that come off of it. Small little processes that were branching ones that we call
dendrites?
Dendrites, exactly. So these are our dendrites. And then we have the big long process that comes off of it. That is the axon. And as we know, that axon can go from the small of our back all the way down to our big toe. It can be like 3 meters long. And we know it ends with those axon terminals and the synaptic end bulbs, little bulbs at the end where we're going to communicate with our target cell. And again, these are the axons. So typically when we are talking about our major structures, even the brain and the spinal cord, basically when we talk about nervous tissue, it is either the part made up of cell bodies or the parts made up of axons. And it turns out a ganglion, and I'm going to go ahead and delete this one so I can start writing again. We define a ganglion as a cluster of cell bodies. All right. Now we need to add one more definition to this. There are, and I guess I have to write this in blue as well. Two divisions to our structural organization. And I think someone already mentioned it earlier. Anybody know the two structural divisions of the central nervous system? Oh, there you go. I gave you one. Central nervous system.
And then I think peripheral nervous system.
And peripheral nervous system. Excellent. Our peripheral nervous system and our central nervous system. Which of these structures we've identified so far make up our central nervous system?
The brain and the spinal cord.
Yes, brain and the spinal cord. So both the brain and the spinal cord make up our central nervous system. Whereas our peripheral nervous system includes things such as the ganglion. So a ganglion is a cluster of cell bodies in the peripheral nervous system. Anybody know what we call a cluster of cell bodies in the brain or spinal cord?
Neuron cell bodies? Or is that?
Well, that is what we're talking about. We're talking about the cell bodies of the neurons. In our brain and spinal cord, a cluster of cell bodies is typically referred to as two ways. We refer to it as our gray matter. Oops, gray matter. There you go. Your gray matter. Or it can also be referred to as what we call a nucleus. Plural being nuclei. So a ganglion is a cluster of cell bodies in the peripheral nervous system. And I guess I should be consistent with my size here. So click that, click that. Excellent. So what do you think our axons form? Let's start easy. What do our axons form in the peripheral nervous system?
Nerves. Nerves.
Yeah, collection of axons in the peripheral nervous system we refer to as nerves. Right, my children get on mine all the time. And how many do I have?
A lot.
A lot, true. And often we can get away with terms like a lot, but let's start easy. Nerves are basically divided into two types. What are the two types of nerves we have in our body?
Visceral.
That's a good way of dividing it, and that's true, but from a functional standpoint. But in this case, I'm talking structural.
Spinal and cranial nerves.
There you go, exactly. Cranial nerves. And now let's be specific. How many cranial nerves do we have?
12.
12 pairs on each side. We together We'll go over the 12 cranial nerves. You will learn everything you ever wanted to know and more about those cranial nerves. And depending on how far behind we get in the section, because we always fall behind in this section, but depending on how much sensory stuff we do, if we don't get to a lot of sensory stuff, then as much as 25% of your lab exam is going to be cranial nerves. So it is a big, big, huge component. Looking at the schedule, it looks like we'll likely get to it somewhere around the 27th. So because we've got some building to do before that, but that is going to be a huge part. And so if you have cranial nerves, what else do you have? I think someone said it, but we'll say it again. Spinal nerves. And anybody know how many spinal nerves we have?
Isn't there like one coming off of each vertebrae to each side or something?
One associated with each vertebrae with one extra thrown in. So you've got the right idea. There's actually 31 pairs. Now, aren't there axons in the central nervous system? Or we say yes.
No.
What do you think we call axons in the central nervous system?
Is that the white matter?
White matter, exactly. Matter, or they can also sometimes be referred to as tracks. You'll see that term as well. Excellent. So there you go. We have cell bodies and axons in the central nervous system and spinal cord, brain spinal cord. We have cell bodies and axons in the peripheral nervous system. But there are a couple more components we want to talk about. One really more specific component we want to talk about in the peripheral nervous system. In our peripheral nervous system, we also have these very elaborate networks of both nerves and ganglia. And these elaborate networks of nerves and ganglia we refer to by the term of a plexus. Oops. Probably one of the greatest examples of one of the plexuses that we have is the enteric plexus. The enteric plexus is actually what we commonly refer to as the gut brain. Right? You woke up this morning and you had breakfast. So you grabbed that nice big huge cheeseburger, and as you shoved that cheeseburger into your gullet, when it got down to your stomach, did you have to tell your stomach, hey stomach, I just swallowed a cheeseburger, it's time for you to do your work?
No.
No. What actually happened is as you swallowed that cheeseburger, your stomach stretched. And it had stretch receptors in the stomach that actually sent a signal, not to your brain, not to your spinal cord, but to this enteric plexus, this elaborate network of nerves and ganglia in the stomach itself. And the stomach actually made its own decision. Hey, I was just stretched out. I need to start churning up the food inside of me. I need to produce gastric juice so I can break that down. So it actually made that decision on its own based on that plexus. Now, again, these are not elaborate, decisions. Stretch means churn, right? It's not deciding where you're going to college or who you're going to marry or anything along those lines. But we do have these plexes that play an important role in helping to make some of the decisions at a local level for many of our organs. All right. Now, here we have our functional organization. Here we have our structural organization. And again, as should be not a surprise to anybody, is there a relationship between these two?
Yes.
Yes. So what functional organization, what function, let's say it that way, does our central nervous system do? Of these three functions, what does our central nervous system do?
Interprets sensory input and
Yeah. Absolutely.
motor input, or is that a different part of the nervous system?
No, absolutely. No, you're absolutely 100% correct. Oops, hold on. Our central nervous system, the brain and spinal cord, has the integration function, where it receives the information, it processes that information, and it makes a decision. All right, hold on. I need to cheat. Come back here. I want to make this blue again for consistency's sake. Excellent. And our sensory and motor functions form our peripheral nervous system, comprised of, as we talked about, the nerves, the ganglia, the plexes, and, or actually you can go plexuses, either are acceptable, and also special sensory structures, like our retina of our brain, or like our olfactory epithelium. So we need to, I guess, throw that over here as well. Special sensory structures. Excellent. And one last vocabulary thing. So let's cheat, and using a green highlighter with a thin line, we can go ahead and draw that division between the peripheral nervous system and the central nervous system. When talking about the peripheral nervous system, information, sensory information that goes in towards our central nervous system. Do anybody know the fancy term we use for going in towards something?
Afferent pathway.
Yeah, exactly. Afferent, which is going into the central nervous system. Oops, that's not how you spell into. Excellent. And so then what we call the motor coming out?
Afferent.
Efferent, exactly. And again, I'm emphasizing that by the way I say it, efferent, which is coming out of the central nervous system. So when we talk about information, we can talk about it as being sensory and motor, or we can refer to it as being efferent and afferent, going into the central nervous system or coming out of the central nervous system. And there you go. Just that easily, we have identified the structural organization of our nervous system, the functional organization of our nervous system, and conveniently enough, put the information together. Because as we know, structure and function go hand in hand. Now, again, we have this done here really nice and pretty, but again, we see the same thing in your textbook and on the lecture slides. We have our sensory information where we're getting the information and sending it to our command center for that integrative function, where we receive the information, process the information, compare it to our memory and make a decision, and then motor going out. And again, I use the classic example of seeing a bear with an axe. Your textbook uses a slightly more boring example where you see a glass of water, process that you're thirsty, and pick up the water to take a drink. But I don't think most people can relate to that. Bears with the axes, everybody understands. All right. Our nervous tissue structurally is made up primarily of two types of cells, neurons and glial cells. We'll talk more about that later. But as we mentioned, the structures of the nervous system are the brain and the spinal cord, our cranial nerves and their branches, our spinal nerves and their branches, the ganglia, our sensory receptors, our plexuses, all of those types of things that we talked about. And again, these structures are typically comprised of either neuron cell bodies, and wherever you have the bodies, you have the dendrites, because they're right there and there's tiny, or the axons. So those are basically the two structures. And again, we'll talk about the anatomy of our neurons in much more detail in just a minute, but typically you have one or the other that form these types of structures, or even within the brain and spinal cord, that as well. And that actually brings me back to one last point I was going to make earlier and I forgot. So luckily I remembered here. Notice when we come back to this, as we just finished talking about, both the brain and the spinal cord are part of this integrative center, which means they make decisions, right? Does that mean your spinal cord decided what you were going to eat for breakfast this morning? Or who you're going to fall in love with, or what you're going to do with the rest of your life? No.
No.
But our spinal cord does make decisions. And what do we typically call the decisions that are made by the spinal cord?
Involuntary?
True, that is one of the ways we would describe it, absolutely, and that would be 100% correct. That wasn't the term I was looking for, but it is 100% correct.
Would it be
Yes.
like,
Impulse?
And say again?
I said impulse.
True, that is how it communicates, but someone give me an example of a decision that your spinal cord might make.
I thought instinct maybe?
Not quite instinct, although you're getting closer with that term.
Reflex.
That's what I was looking for, a reflex, absolutely. If I put my hand on something hot, I pull away from that before I'm even aware of the fact that I should be cursing, right? Why? Because that information went into my spinal cord received that information, my spinal cord made a decision, my spinal cord sent a signal out to my arm to move my hand away before that information ever got to my brain. So absolutely, our spinal cord does make decisions, and those decisions that are made by our spinal cords, we call reflexes. Because you're right, they're involuntary, we don't have to think about them, right? But they're there and they're vitally, vitally important. So it absolutely does make decisions. We think of the spinal cord as just carrying information to and from the brain, but it does a lot more than that. Excellent. We talked about, where are we? Excellent. We talked about our central nervous system, that integration and command center, the brain and spinal cord, our peripheral nervous system, carrying information, made up of those nerves, the ganglia, the sensory receptors, the plexus. And we talked about how we have sensory, which is afferent, and motor, which is efferent. But we also mentioned that there are subdivisions to these afferent and efferent pathways. For sensory, we have our somatic sensory, general sensory information, our autonomic or visceral sensory, made from those enteroreceptors that we talked about, and our special receptors, which give us that specific, unique information like sight or hearing. Motor, we have our somatic motor, which controls our skeletal muscle, our autonomic motor, which controls our smooth muscle, our cardiac muscle, and our glands, and are further subdivided into our sympathetic and parasympathetic branches, our fight and flight, and our rest and digest. And notice your book has this great picture that almost does a perfect job of showing this. Notice it gives us a great example of our central nervous system, here with the brain and the spinal cord, our peripheral nervous system with the afferent and efferent pathways. Notice we have an efferent pathway coming out to our skeletal muscle, our somatic motor. Our autonomic goes to things like smooth muscle or cardiac muscle. And there's the two branches, sympathetic and parasympathetic. And we have our sensory coming in, our visceral sensory with those enteroreceptors telling us about the pH in our stomach or the stretch of our stomach or something like that. Our somatic sensory here from the skin. And notice the one thing we're missing here is we need an eyeball. And with this eyeball, we could then have our special sensory. Oh, let's pick that color. Carrying information in as well. So it's missing our special sensory. And then it's a perfect picture from our textbook. All right. Questions on that? All right, excellent. So now that we understand the functional and the structural organization of our nervous system, we need to talk more about our nervous tissue. As I mentioned earlier, our nervous tissue is primarily made up of two types of cells, neurons and neuroglial cells. So we're going to need to spend some time talking about that. We got a lot to talk about today, so this is a good natural stopping point. Let's go ahead and take our first break here. Looks like it's 1 o'clock, so we will restart at 1:15, and I will start the recording again at that time. All right, excellent. Any questions before we take our first break? All right, then I will see you guys in 15 minutes. All righty, let us go ahead and get started. Any questions before we dive back in? All righty, our job now is to then talk about nervous tissue. As I mentioned, nervous tissue is primarily made up of two types of cells, neurons and neuroglial cells, or what are also known as glial cells. These terms are glia are interchangeable. Glia and neuroglial are both interchangeable terms that you are welcome and encouraged to use. Neurons are the ones we've been talking about the most so far up to this period of time. These are typically the functional cells. These are the ones that are responsible for producing and sending the action potentials. These are the ones that I said, some of them can be over 1 meter long with their axons, carrying information to all different parts and places in our body. Whereas the and again, these are similar to this kind of what we saw in muscle tissue, in that neurons are irritable. What did we say it meant to be irritable?
Electrical stimuli.
Yep, you got the right idea. To be irritable means that they receive a stimulus and respond to it. And you're right, they respond to that stimulus by producing a big positive electrical signal, right? So they produce an electrical signal. And that electrical signal, because they produce an electrical signal, we also said that like muscle tissue, they are electrically excitable, right? They also have conductivity. What do I mean by that?
Conductive.
Yeah, but what does it mean to be conductive? Normally, see, again, I have to work on some of my analogies because I'm sitting here. Normally, I'm sitting here in the classroom, and in the classroom, I have this big, huge wooden yardstick that at the end has these two little metal ends. And what I like to do is grab that yardstick right in the middle, and right at this period of time, I will then shove that yardstick into a light socket. And it sparks and smoke comes up and all those types of things occur. However, am I at any risk when that occurs?
No, because the wood isn't conductive, but if it were metal,
Exactly.
it would be.
Exactly. The wood is not conductive. So while the metal at the end can cause the sparking and the arcing in that light socket there, it can't travel up the wood, right? But you're absolutely right. If instead it was a metal yardstick and I did that, then what was left of my hair would stand up on end. Absolutely. So conductivity means that it can spread the electrical signal along its length. All right, so again, as we talked about, some of these can be as much as a meter long. So that ability to be able to produce that electrical signal, and not just produce it, but spread it long distances, is a vitally important process. All right, so hugely important cells, these neurons, but they can't do the work by themselves. The neuroglial cell, and there are dozens of neuroglial cells that have been identified in our nervous tissue. I'm going to only hold you responsible for identifying a very, very small portion of them, but there are dozens and dozens of these neuroglial cells, and their job is primarily to help the neurons do their job. They're the support cells. However, if you notice, there's a little asterisk by that. How many of you have heard of an individual by the name of Albert Einstein?
Nah, sounds familiar.
Okay, one or two people may be some familiarity with it. Good news is, incredibly smart individual. Bad news is he is now dead. So again, if you didn't get a chance to meet him, then too bad. But when he died, incredibly smart individual, absolutely incredibly smart individual. Yes, he was the light bulb guy. But when he died, he gave his brain to science. And all the neuroscientists were all a slather to be able to get a look at his brain and see what kind of unique neural connections his brain made that us mere mortals aren't able to do with that. Yes, I know it was not that he was not the light bulb guy. He was the guy with the kite and the lightning bolt. But the point is that these neuroscientists got a hold of his brain finally and were able to look at the neural connections. Yes, that was Ben Franklin as well. Excellent. You guys know your history. Great. Let's get to the point. The point is, neuroscientists finally got a hold of his brain and wanted to look at the organization of the neurons in his brain that allowed him to make such tremendous leaps in knowledge and understanding that, you know, we are not able to do. And what they found out is that actually his neural network is pretty much the same as everybody else's. So in fact, you can call grandma this afternoon and say, hey, grandma, guess what? I have the exact same neural network as Albert Einstein, and she'll be incredibly impressed and send you 20 bucks in the mail. Now, it didn't mean that his brain was identical to a typical brain. What they actually found is that some areas of his brain had as many as 4 or 5 times as many neuroglial cells as a typical brain would have. So while we think of and know that neuroglial cells do play a role in support, should you take this class 5 or 10 years from now, and quite frankly, I don't know why you would, but if you did, as our understanding of these neuroglial cells grow, we may find out that they play a much more important role in our cognitive function and understanding how our brain works. But for now, we'll stick with the company line. We definitely know the things that it does to help us support our nervous tissue, and so we will focus on that for now. All right, so let's take a closer look at these two types of cells, starting first with a neuron. And here you can see your friend and mine, that neuron, looking pretty much like we talked about and drew it earlier on. Do I have Greek mythology examples for the day? I'm not sure if I have one for today. We'll work on that. Let's go to the pretty pictures. So let's talk about the anatomy of a typical neuron. We've already kind of seen some hints at this, but let's be a little bit more specific. As we know, there is a, let's not use green. There is a big, large, centrally located cell body. And the cell body is referred to as the soma. As we expect inside of this, of course, is going to be a nucleus. So remember, that is somatic. Somatic is, again, soma is an adjective that means body. So somatic motor neurons are the type of neurons that control that, but each neuron has its own cell body, or what we refer to as soma. So they're using it the same way as it refers to the body. And of course, as we know, in the nucleus, you will often see dark spots where transcription of the DNA is taking place. And what do we call those dark spots found inside of there again?
Nucleolus.
Yeah, nucleolus, or plural because I've drawn two of them. Nucleoli. Excellent. So those are the kind of things that we expect to see inside of the neuron cell body. However, when they looked at it, cell body wasn't identical to what we expected. It turns out when they looked at this under a special microscope using a special stain, what they found is inside of the cell body was this massive amount of very dark stained material. This particular material happened to show up when they used a special stain called a Nissl stain. So these things were referred to as the Nissl bodies, because this cell was just packed filled with this really, really darkly stained material. But then as our microscopes got better, what they started to realize is that this really darkly stained material that was practically filling our neuron was all these massive interconnected flattened membranous sacs with all these globular components on the outer surface. What does that sound an awful lot like?
Rough endoplasmic reticulum.
Exactly, and it turns out that's what the Nissl bodies are. The Nissl bodies are rough endoplasmic reticulum. And someone remind me again, what is it that the rough ER does?
Don't they package and ship
Well,
ship?
they're not the packaging and the shipping part. Something has to happen before the packaging and shipping.
Oh, they synthesize proteins? 'Cause
Yeah,
that's what
exactly. This is what synthesizes proteins. And so not surprisingly, our neuron is chock filled with a lot of proteins. Some of those are structural proteins that are going to help to give structure and support. After all, if this cell is going to have a process a meter long, it needs some structure, it needs some support to it. But remember, our neurons, while they produce big electrical signals, how do they communicate with their target cells?
through
the
electrical.
Yeah, they release a chemical signal. What's the general term we use for that general signal?
An
A
impulse.
neurotransmitter.
Produce neurotransmitters, exactly. So by having a big, huge, massive endoplasmic reticulum, these cells are actually able to produce the neurotransmitters that they need. Yes, question.
Yeah, is this the same neuron that stimulates muscle contraction, or is this a totally different thing?
These are all neurons. This is a typical neuron. So obviously those include those, but there are other types of neurons as well. So
Okay.
this is we're not talking about motor or sensory or anything else right now. Right now, we're just talking about the anatomy of a typical neuron. Now, of course, what's the key word in that sentence?
Typical.
Typical, because typical means that they're not all going to be this way, but the majority of neurons are going to look just like this. Absolutely. Because they're all fairly similar in their function. And we'll actually see the differences. Once we have the basic anatomy, we can see how that basic anatomy is modified to form the other types of neurons. All right. Go ahead.
Is there a difference between the Nissl bodies and this chromatophilic substance that the book talks about?
I'm not familiar with that. Where does the book talk about that?
Never mind.
Yep.
Ignore that.
Okay.
Just kidding.
Okay. I'm not going to say no. That's not a term that I've heard, but it could. I mean, again, this was called Nissl bodies because of the particular stain, right? So basically it's an artifact, and that's basically chromatic refers to color. So that could be. I'm not familiar with that term, but I would have to see where.
Okay. I don't know. It was underneath it. It says structure of a motor neuron, but that's not what we're talking about, is it?
Well, we are not specifying motor yet, but
Okay, that's what I thought.
but again, since we're talking about typical neurons, it's not uncommon for them to have these things.
Okay.
Okay, there is two other things that we need to talk about. As we know, our neuron also has two types of processes. And those two types of processes are going to be our dendrites and our axons. All right. So a typical neuron has many of these, whereas a typical neuron has only one axon. These dendrites are short and typically very branching, whereas our axon is long and unbranching. So if we were to draw this here, we would draw some small little processes coming off, and these would be our dendrites. Oops, that didn't need to be quite that elaborate, but it's all right, we get the idea. And our axon, which we'll draw a little bit more elaborately. So let's cheat. All right. There's really two more big differences between our dendrites and our axons. Dendrites receive information, whereas axons send information. And of course, when we talk about that axon being able to send information, send that signal, our axon is the only part of the neuron that can produce and propagate. Propagate, remember, is just a fancy word for send. The action potential. What that means is that over here, the dendrites and the cell bodies cannot produce an action potential. It only occurs on the axon. Now, if you remember correctly, the plasma membrane of our muscle cell was able to produce and propagate an action potential as well, because it had a special plasma membrane, and we gave that special plasma membrane a name. What was the name of the special name for the plasma membrane of a muscle cell?
Was that the sarcolemma?
Yeah, sarcolemma, absolutely. So, not surprisingly, the special plasma membrane of the axon that allows it to produce an action potential we call an axolemma. All right, so our axon's plasma membrane is called the axolemma because it is specialized, because it is able to produce and send an action potential. So notice if we come back here to our cell body, we can basically form a line right here. All right, this part back here, the cell bodies and the dendrites the cell body singular and the dendrites plural cannot produce an action potential. The axon can. However, do you think the axon right here is right next to that line is able to produce an action potential?
Yeah.
Do you think the transition is really that abrupt, where suddenly you have
No.
plasma membrane that can't do it, and then suddenly you have plasma membrane that can?
No.
Turns out it's not that abrupt. Instead, there is a transition area. This transition area basically is where we can think of it in two ways. This is where the axon connects to the cell body. And this is the transition area to the specialized plasma membrane. And of course, we're going to give a special name to this special connection location and this special transition area. And what we call it is the axon hillock. So if I do that and I do that, I can fit it all into that space. That works. All right. You with me so far? Okay, let's draw some arrows.
I have a quick question.
Yes.
Did you say only the axon is the only part of the neuron that can propagate and send an action potential?
Correct. That is what I said. Okay. So let's draw some quick arrows. Arrow 1 right here at the very end of the axon hillock. Do you think we can produce an action potential there?
No.
Really?
Maybe.
Yeah, I think they're right next to the regular standard specialized axon. Absolutely. However, as we talked about before, right here, right at the beginning of the axon hillock, do you think you can produce an action potential there?
Yes.
No. So again,
No.
remember this. So think of it this way. This is a transition area, right? Think of it in terms of like a pool. There is the shallow end and there's the deep end. And typically the change isn't abrupt. There is a gradual change until eventually you reach some critical point where you can no longer stand, right? And as long as you keep going deeper, you still cannot stand. But you're able to walk, you're able to walk, you're able to walk, getting deeper and deeper and deeper. And then suddenly the transition has happened enough where suddenly you have to swim because you cannot stand anymore. All right? Does that make sense?
So it basically takes the whole axon hillock or whatever to
It
produce an
takes a
action
portion of the
potential.
axon hillock. And that's really the key. During this transition, there is going to be a point, and that point is going to be somewhere here in the middle. It isn't a line we can actually see. You know, it isn't a structure we can actually see, but we know somewhere here in the middle is going to be an area. So let me get rid of these two arrows. This, and put this one here instead. This portion right here is a location where the axon has changed enough to be able to form an action potential. Right? Before that, it hasn't changed enough. After that, it's definitely able to do it. But this is basically the first location where the axon has changed enough to form an action potential. And this small region inside the axon hillock is called the trigger zone. So this trigger zone is located inside the axon hillock. Where the axon has changed enough where it can form its first action potential. All right. Questions on that? All right. Remember I said the Nissl bodies were, or now we know the massive amount of rough endoplasmic reticulum that it has is one of the specializations of the cell body, but I did say there were two. As I've talked about, some of these axons can be up to 1 meter long. And so as we talked about, there are proteins that need to provide structure. We know that cells have cytoskeleton, and that cytoskeleton helps to maintain the shape and the structure of them. Some of those cytoskeletal structures will help to maintain the shape of the dendrites, but a massive number of these cytoskeletal structures are going to help to form the axon and are going to form pathways that our neurotransmitter can be used to travel down the axon. This is the cytoskeleton, right? These are protein fibers inside of the nervous cell. And so not surprisingly, they give them fancy names. Oops, shoot. I hate it when I do that.
Wait, so Professor, the green lines that are like going down the axon, that's the trigger zone?
No, that's why I'm changing this right now. The trigger zone is the red part.
Oh, okay.
There we go. No, these green structures that I have drawn represent our cytoskeleton, right? Proteins that help to maintain the shape and the structure. And allow for transport. If you remember way back in the day when we first talked about the cells, we talked about how the cytoskeleton, there could be motors that move along them, carrying and transporting vesicles and things like that from one end to the other. So in this case, these structures can help to allow that neurotransmitter to be transmitted down to the end of this. And these, because they are protein fibers inside of a muscle cell, of a nervous cell, are called neurofibrils. So they're just cytoskeletal proteins that give the axon and the dendrites shape and structure, and can be used for transport. Yes, correct. The red part, the red region is indeed the entire axon hillock. And then in the axon hillock is where you find the trigger zone. All righty. That's kind of everything that's going on in the proximal end. The good news is we know exactly what is happening at the distal end. At the distal end, we know that our single axon can branch. And when it branches, those branches we call the axon terminals. We know at the end of the axon terminals are an enlarged structure. And what do we call that enlarged structure?
Synaptic end bulb.
Excellent. And this synaptic end bulb has two specializations to it. What are the two specializations of our synaptic end bulb?
Voltage gated calcium channels.
Perfect. We know that it is going to have voltage gated calcium channels. And what do we need those voltage gated calcium channels for?
To release the acetylcholine inside the vesicles.
True, although remember, acetylcholine is just the chemical that is used at, well, is one of the common, but at the neuromuscular junction. But we had a generic term for that as well. What was the generic term?
Exocytosis.
Now that's how we release it. What acetylcholine is a specific type of a chemical signal. And what is that term we use for the chemical signal?
The neurotransmitter?
Neurotransmitters. Absolutely. Acetylcholine is one of the most common, but we'll just use the generic term acetylcholine for right now. So here are our vesicles containing whatever the neurotransmitter is. And of course, we need those voltage-gated calcium channels to open to let them out as well. Excellent. Did I see a hand?
Yeah. So in this case, it wouldn't be acetylcholine that would be released. It would be the other proteins that are inside
It
of
could
it.
be. There are over 100 neurotransmitters that have been identified in the human body. Okay. Acetylcholine is absolutely one of the most common. So if you were to randomly reach into the body and pull out a neuron, it might be acetylcholine. It most likely will be acetylcholine, but are we 100% certain it's going to be acetylcholine? No. But what we are 100% certain is that it contains a neurotransmitter, all right? Because that's the generic term for that chemical signal. So you're right. Absolutely one of the most common ones, and the most common one we're going to talk about is acetylcholine. It is by far the most common in the peripheral nervous system that we have. But there are dozens upon dozens of other ones as well. So at this point, since we're just talking generic, it's best to use the generic terms like neurotransmitter. But you're absolutely right. The most common one is definitely acetylcholine. All right. I have done truly an amazing job of drawing this, but your book's got some pretty pictures as well. So let's take a look at that. Notice again, here is the cell body, the soma. Here, are those Nissl bodies. And again, at first they didn't know what they were, but now that we look at them, we see they're flattened membranous sacs with lots of dark structures around them. So that is our rough endoplasmic reticulum. Notice we can see, and I'll use red for pink for this one. We can see the protein fibers, the neurofilaments that are helping to form the cytoskeleton, that are forming giving the structure and the integrity to it. Here we have the many short, elaborately branched dendrites that receive information, and the one singular axon that carries information away from the cell body. Oops. Notice here we see that transition region that is the axon hillock. And notice they haven't drawn it here because it isn't something we can see anatomically, but we do know somewhere in the middle of this axon hillock is going to be where we're able to produce our first action potential, and that is the trigger zone. All right. So that's everything we needed to see in the anterior part, proximal part. And as we know, here's what that distal part looks like. Our single axon branches into axon terminals. And notice again, as we said, there are many terms for this. Your book uses the term teledendria. That is fine. We use the term synaptic end bulbs. Your book uses the term synaptic terminals, terminal boutons, synaptic buttons. There's all sorts of things as well. And we're using the term synaptic end bulb. Yes, the two specializations that, if you remember, we found in that synaptic end bulb is voltage-gated calcium channels that control the release of the neurotransmitter. So it's the vesicles containing neurotransmitter and the voltage-gated calcium channels that allow it to be released. Absolutely. So there you go. We've already seen the distal end of this. Now we've identified the proximal end of that, and that is the basic anatomy of a typical neuron. All right. Yeah, teledendria is just their term that they use for the axon terminals, the branches of the axon. So teledendria, axon terminals, synaptic end bulb, synaptic terminals, right? Synaptic boutons, terminal boutons, synaptic buttons. I mean, like I said, there's a ton of names for these things. Excellent. Well, it is Thursday, so what you do with your weekend is completely up for you. All right, excellent, excellent, excellent. So That is a typical neuron. However, we keep using that term typical, and that means that there must be specific types of neurons. And not surprisingly, when we talk about specific types of neurons, we can categorize neurons in one of two ways. What do you think the two ways we can classify neurons are?
Let me
Their
guess.
size and shape.
Exactly. Shape and function. So we have structural classifications. And as it turns out, there are 3 of those. And we have functional classifications. And there are 3 of those. Now, I'm going to guess functional are probably easier to understand based on the functional organization of our nervous system. What do you think the 3 functional classifications of our neurons are going to be?
What we went through at the beginning, the sensory, interneuron, and motor.
Okay,
Or-
sensory, absolutely. Motor. And then what would the third be?
Somatic.
Well, not somatic. What was the third function when we were doing our classifications of the nervous system? Absolutely. It was that integration that took place in between. And in this case, that integration, absolutely, this third functional classification are what are known as interneurons. Right? And when we think of them in terms of their function, what do sensory neurons do?
Sense things.
True. And what do they do once they sense that information?
Send it to the brain.
There you go. And what was the special term we used for going towards the brain?
Integration system.
Afferent.
Exactly, there. Afferent.
Afferent.
And again, as long as you spell it right, I don't care how you pronounce it. I do the afferent to emphasize the difference between afferent and efferent, which are the motor. All right, remember these carry information out of the, and actually we should be more specific. I'm going to change that, out of the central nervous system, and they're efferent.
Can we use input and output instead of those words, or?
Yes, you can use the terms into and out of, that is fine, but definitely you need to know what those terms mean. If I ask you what type of information is afferent, you need to know that that's sensory. So again, you're welcome to describe them however you want, but you definitely need to know the definitions of these terms.
No, I was saying input and output. Sorry. Or is it the same?
It's a good question. So here's what I would say. Technically, it's not correct, but in this circumstances, it's probably okay to use. Again, like we've talked about before, everything we talk about in this class is more complicated than we're talking about. So, but yeah, I mean, so carrying information incoming information, input probably be fine. Output for carrying information out of the central nervous system, efferent coming out. And then our interneurons, basically, since one's carrying into the central nervous system, one's coming out of the central nervous system, the interneurons are basically the ones in between. These are located within the central nervous system. All right. And they basically process information between the sensory and the motor. So those are our 3 functions. And I think those are pretty simple and straightforward. I don't think there's any surprises there. And we pretty much were able to guess that. So let's talk about our structural classifications. the first structural classification, and let's use color for this. For our first structural classification, we have a centrally located cell body. From that centrally located cell body, we have one dendrite. One dendrite that feeds into it. Now that dendrite can have branches coming off of it, so it can receive a lot of information. But it has one dendrite coming into the cell body, and it has one axon coming out. And again, as we know, it can branch into those axon terminals. So notice we have our centrally located cell body. We have one dendrite. We have one axon. It shouldn't be a surprise. We always know we have one axon. And of course, as we can also tell, this is carrying, information in this direction. So notice our information is going from the top of the page down to the bottom of the page through this neuron. Now, if you notice, this neuron off of its cell body basically has two processes that come off of it, one dendrite and one axon. And because it has two processes that come off of it, we call this a bipolar neuron. Let's talk about our second structural classification for a neuron. It also has a cell body. Well, let's go back for a second. This was a centrally located cell body. Because that's what it has. It just has one. Again, remember, why is one of those questions we can't always answer, because again, the engineer who designed all this didn't leave us our cliff notes.
But it looks like it has many dendrites on it.
It doesn't. The same way you have one axon that can branch at the end, the same thing with the dendrites. The dendrites can have many branches that come together into one trunk. And that one trunk then carries it into the central body. Notice, again, it's about what connects to the body. Two processes connect to this body, one dendrite and one axon. So it is a bipolar neuron.
So it's just for this specific type of neuron? Because the other ones I thought we were drawing had like a bunch of little dendrites.
And remember, we called that a typical neuron, but typical means not all.
Gotcha.
So in this case,
thank you.
This specific structural classification is bipolar neuron.
And this is one of the three structural classifications.
Correct. Right.
Got it. Thank you.
Yep, no worries. You guys are getting it. I like that. All right. I need green again. We have a second structural classification, which also has a cell body. But in this case, it is a peripheral cell body. It's a cell body that's off to the side. And what happens is a single axon connects to it from the side. Again, axons can branch and have axon terminals that allow it to communicate to many things. And of course, it has to receive information. So it still can have dendrites, but the dendrites, notice, don't connect to the cell body. In this case, the dendrites connect to the axon. So if you think about it with this one, how many processes actually connect to this cell body?
Just one.
So what do you think we call this neuron?
A unipolar.
There you go. It is a unipolar neuron. Notice it carries information the same way, from the top of the screen to the bottom of the screen. Receives it at the dendrites, sends it down the axon. The only difference is here that its neuron is to the side. Now, lastly, notice neither of these look like the neurons we've been drawing up to this period of time. The neurons we've been drawing up to this point of time have a central cell body. This one has two or more. Oops, that's not or more. 2 plus, there we go. Dendrites that are going to connect to it. So I need my blue for that. And of course, oops, uh-oh, come back. There we go. It is going to have one axon, just like before, and that axon can branch. This is what we've been drawing as a typical neuron. Now, if two processes we call bipolar, one process coming off the cell body we call unipolar. If you have basically three or more, guess what we call this one?
Multipolar.
Exactly. This is a multipolar neuron. And as you guys have also very clearly guessed, which of these three types are the most common structural classification? Oops.
Multipolar.
Multipolar, exactly, right? Because this is the one we've been drawing the most when we've been talking about it. So absolutely, this is the most common structural classification. All right, excellent. Of our three functional classifications, which one do you think is the most common?
The bipolar.
Well, functional. When we think functional, which of our functional classifications do you think are the most common?
The sensory.
Well, so think of it this way. Where do you think we have more neurons? Carrying information into our central nervous system, carrying information out of our central nervous system, or actually inside our central nervous system?
Inside.
Inside, absolutely. In fact, about 90%, over 90% of our neurons. Let's do this in, what's a color we haven't used yet? I don't know, orange. So our most common functional classification is our interneuron. And let's go ahead and do this too. Most common structural classification is a multipolar. So I need to, oops, move this first. I can erase that. And then I can move that up here. All righty. With me so far? All right, stunned silences say yes, so that's perfect. So let's take this one step further. Do you think that there is a relationship? Oops, no. There is a relationship between the structural classification of our neurons and the functional classification of our neurons?
Yep.
Yeah.
Yes.
Absolutely. So it turns out that most of our sensory neurons are unipolar. So we can write that over here as well. Excellent. Of course, what does most mean?
Not all.
Not all, right? There are a few found only in the special senses. So this includes, again, things like vision, things like hearing, things like smell and taste. There are a few of these that are only found in the special senses that are bipolar. So bipolar form are found in some of our special senses. So most of our sensory are unipolar. A very few, only involving things like I said, like hearing, like sight, like smell, like taste, where you'll find bipolar neurons. And so then notice, not surprisingly, our interneuron, which is the most common, and our motor are both multipolar. So our multipolar neurons form our motor and our interneurons.
So all motor are multipolar?
Yep. Well, again, all is a sky is blue kind of answer, but for our purposes, yes. All motor are multipolar, all interneurons are multipolar.
Thank
you. Yep. And again, we've done this here with the pretty pictures that I've drawn, but we've got these great illustrations from your textbook. Again, notice when we talk about the structural classification of neurons, as we saw, it is based on the number of processes that come off the cell body. Here we see an example of that bipolar neuron with one dendrite and one axon going smack dab through the middle of our neuron. And notice its function is in special senses, in our retina for vision, in our inner ear, in our nose for olfactory area, and so on. Here we see an example of a unipolar. Notice its cell body is located off to the side and just a single axon travels next to it with dendrites at one end. So again, information is still going down the screen on both of these. And like we said, our unipolar neurons form most of our sensory neurons. And there's our typical neuron that we've seen before with multiple processes coming off of it. So it is a multipolar neuron, and it forms our motor neurons and our interneurons. And we can do the flip side of this as well. Like we said, our functional classification is basically the direction they carry the information. Motor carries it out of the central nervous system. It is efferent, and those are multipolar. Sensory is afferent, coming into the central nervous system, and those are mostly unipolar, and again, with a few bipolar. And we have our interneurons, making up 90% of all the neurons in our body, basically found in the central nervous system between the sensory and the motor neurons. and they're multipolar. So notice, if you were to throw all the neurons in your body into a bucket and reach in and pull it out, it would most likely be an interneuron that you would grab if you reached in there randomly, and it would most likely be multipolar. All right. But even that, do you think we just have one type of multipolar neuron?
No.
No, of course not. There are dozens of different types. Here are two of the more extreme examples. Notice these are both multipolar neurons. Both have multiple dendrites coming out of them and a single axon. This one over here on the right, as you can see very clearly, is called a pyramidal cell because it has a pyramid-shaped body and these long processes. This one over here, notice, has this huge, elaborate fan-like dendrites. And what's really cool is, notice it looks almost like our artist wasn't very good, that they just drew this fan two-dimensional. Well, it turns out it really is two-dimensional. It's not like a tree where the branches go anywhere. It really is like a fan. If you take this cell and rotate it to the side, all the dendrites all line up on the same plane. It is this big, huge, very linear fan-shaped cell. So, of course, they call these fan neurons. Wait, they don't call them fan neurons. What do they call them? No one wants to pronounce that name. Purkinje. They're Purkinje cells. Any idea why they're called Purkinje cells?
Bob Purkinje.
Absolutely. Good old Bob Purkinje. Purkinje was a truly amazing neuroanatomist. He loved the microscope, and he was an amazing artist. If you ever get a chance to look up some of his illustrations that he did, he was a truly amazing artist. And so he loved to take parts of the body, tissues from the body, explore them under the microscope, and draw the things that he saw. Some of the findings that he found were truly remarkable and truly amazing from an anatomical standpoint. And he was an incredible anatomical whore. So pretty much anything he drew, he put his name on. So here, these Purkinje cells happen to be found in our cerebellum, located in our brain. When we get to the heart, you'll learn about Purkinje fibers in the heart. There are other places where we're going to find Purkinje cells. They're all over the place. He stamped his name on everything. Nerves you can see with the naked eye, but you can't see in some nerves you can see with the naked eye, but you can't see the individual axons typically. No. Was there another hand with another question?
Yeah, you kind of answered it. I was gonna ask about like Purkinje fibers and stuff like that because I've heard of Purkinje fibers, but
Yeah,
not
Purkinje fibers are found in the heart, and it's the same guy. He drew those fibers in the heart and named them after himself. He drew these cells in our cerebellum in our brain and named them after himself. Yep, he was quite a character. And again, we keep emphasizing this relationship between structure and function, but we can't forget location. And here's a perfect example. As I just mentioned, these Purkinje cells are found exclusively and only in a part of the brain we call the cerebellum. It's that big knob, a knuckle type thing on the back and the bottom of the brain. If you haven't seen it, it's okay. We'll take a look at it later. Whereas these pyramidal cells are found exclusively or almost exclusively in the gray matter of the cerebrum. Basically, this is most of our cognitive brain, most of the thinking and processing and touching and feeling and seeing and things that we do are these connections of these cells up here in our nervous tissue. So like most things we talk about, there's far more complicated examples of these, but so there's not just one type of multipolar and one type of unipolar and so on and so forth. There are lots of different types, but these are the main categories. All right. And again, notice as I just mentioned the beauty of this. This cell, which we can easily recognize as that Purkinje cell, we now know its structure. We look at it, we see it is a multipolar neuron. And as we know, these things go hand in hand in hand. Purkinje cells are found in one location and one location only, cerebellum. Cerebellum happens to be in the central nervous system, so functionally, guess what type of cell this Purkinje cell is? What do you think functionally it is?
Interneuron.
It's an interneuron. There you go. Structure, multipolar. Function, interneuron. Location, cerebellum. Location, structure, function all go together. And so again, it's going to be one of those things that we are going to emphasize dramatically. All right, excellent. Your book's got some nice tables that, again, talk about these different types, multipolar, bipolar, unipolar, where you find them, what they do, what they look like, yada, yada, yada. All right, questions on that? All right, we have done the anatomy of the neuron. Let's switch gears to our glial cells, or neuroglial cells. Looks like this is another good stopping point because, like I said, we have a fair amount of material to cover today. So it looks like it's 2:15 right now. Let's go ahead and take our next break. We will start at 2:30. And at 2:30, we will pick up the lecture from there, and I will start the recording at that point. All right, and it helps if I can spell recording. All right, excellent. Any questions before we take our next break? All right, I will see you guys in 15 minutes. So we have talked about the anatomy of the neuron. We'll talk about the function a little bit later. But we've talked about the anatomy of that. I want to talk about our support cells, those neuroglial cells. There are dozens and dozens of neuroglial cells that have been identified in both our central and peripheral nervous system. And they are distinguished by different ones in the central nervous system and the peripheral nervous system. We are going to focus on the 6 most common of these. 4 of which are found in the central nervous system, and 2 that are found in the peripheral nervous system. Again, as we talked about, these may serve many other important cognitive functions as well, but for now we will focus just on the basic support functions of these. And of course, here's the pretty picture showing the 6 different types of neuroglial cells. 4, as I mentioned, we'll talk about in the central nervous system, and 2 we will find in the other. All righty. So let's talk first. We will start first in the central nervous system. And we will talk first about astrocytes. Astrocytes are by far the most common type of glial cell found in the central nervous system. And here you see kind of an illustration of what one looks like and why it would get its name. Notice it has a centrally located cell body, and then it has a massive number of processes that come off of it so that it looks kind of star-shaped, hence the term astrocyte. It has this star shape to it. This star-shaped structure allows it to do a couple of things. First, it helps to form the framework of the nervous tissue. Basically, it is the scaffolding that is going to help to hold the nervous tissue together, give it some shape, give it some structure. If we were in the classroom, we would actually get a chance to do a brain dissection. And when we do that brain dissection, we're doing it on tissue that has already been fixed. So basically, they have kind of the feel of like a rubber ball to them. The actual nervous tissue itself, before it's fixed, is much softer, has kind of the consistency of like a Jello jiggler type of consistency to it. And so it needs some structure, it needs some integrity, and that is one of the main things that the astrocytes do. But the astrocytes also have a lot of other important functions as well. Those include regulating the ionic concentration, of the tissue. In particular, it plays an important role in absorbing potassium. As we know, there should be a large amount of potassium inside the cells and only a little bit in the interstitial fluid surrounding it. So it's going to help to absorb calcium and get that out of the way. It also helps to reabsorb and remove neurotransmitters. If you remember when we talked about our neuromuscular junction, for instance, one of those synapses, we have that synaptic end bulb, we have that motor end plate that it's talking to, and we know that that neurotransmitter, when it is released, one of the things we talked about is that it can diffuse away from that synapse. And when it's away from the synapse, it can't affect that synapse, but it could also just as easily diffuse back, or it could diffuse to another synapse. So we don't want these neurotransmitters floating around all over the place. So by being able to have these cells to help to break down and reabsorb these neurotransmitters, they can stop it from affecting cells we don't want them to affect. All of those are super, super vitally important functions, but it isn't the most important function of these astrocytes. So notice, as you can see, our astrocyte here, again, has these processes that are kind of holding things in place and stabilizing it. But if you notice, if you look closely, you can see that many of its processes are wrapping around these two red things. Anybody have any idea what these two red things might be?
Capillary.
Yeah, if you weren't sure, you could just read it right off the screen right there. Absolutely. These are capillaries. What these astrocytes do for us is they help to form what we call the blood-brain barrier. Our blood-brain barrier plays a vital role in regulating what gets out of our blood into our nervous tissue. As we know, blood carries lots of important things in it. Oxygen, nutrients, right? Gases that we need, all chemical signals, all of those types of important things. But it also carries wastes. It also carries lactic acid. It also carries all this bad stuff, toxins and things like that as well. And do we necessarily want things like toxins or harmful chemicals getting out of our blood vessels into our nervous tissue?
No.
No, because those things could damage our neurons. But that's okay. If one of our neurons die, we can just grow a new one, right?
Nope.
No.
No, exactly. Remember, our nervous tissue doesn't regenerate. So we want to be protective about what gets out. We want to limit what is able to get out. And that is that vital function of our blood-brain barrier. It helps to limit the types of materials that are able to get out of our blood into our nervous tissue. And it helps to protect our nervous tissue. Now, is it perfect? Well, I'm asking the question, so what's the obvious answer?
No.
No.
No, right? Because after all, if you think about it, all this cell is doing is taking its own plasma membrane and wrapping it around the capillary. Plasma membranes are primarily made up of phospholipids, which are, of course, lipids. So something that was lipid soluble, like, oh, I don't know, alcohol, would alcohol be able to easily pass across this blood-brain barrier and have some type of impact on our nervous tissue?
Yes.
Yeah.
Oh
Yeah,
yeah.
absolutely. Yes. A little too much enthusiasm there, but absolutely. Yes, absolutely. It can. And so that is, so again, it's not going to be perfect, but it is going to play a very important role in helping to limit what can get out. Now, here's the other important thing, and I think we've talked about this in other areas, but this is especially true in our nervous tissue as well, right? Knowledge is power. The more we understand something, the more we're able to use it to our advantage. We talked about that with botulinum toxin, how, you know, it went from being this horrible, horrible thing that was found in spoiled cans, that now we're injecting it into, you know, muscles to relax them, for turned eyes, into sweat glands so that they don't sweat excessively, right? All these types of important, and of course, all the cosmetic, yes, celebrity faces as well, all the cosmetic uses as well. But as we learn how the body works, we can use that to our advantage. And this is another one of those really, really classic examples. Pretty much from the second we learned about the blood-brain barrier, scientists, and in particular pharmaceutical companies, have been trying to find ways to get around this blood-brain barrier. One of the classic examples of this involves a neurological disorder known as Parkinson's disease. Anybody here heard of Parkinson's disease before?
Yes.
Parkinson's disease is a degenerative neurological disorder. And one of the common symptoms, well, there's two common symptoms, there's many symptoms, but two of the very common stereotypical symptoms is when a person is at rest, they have a resting tremor. So like when their hands are standing by their side, their hands will be tremoring. When they're moving their hands, they don't tremor, but when they're at rest, they have this resting tremor. The other thing is they have difficulty starting and stopping movements. So if they're reaching out to grab a glass of water, they are not able to always do it smoothly. They'll be very jerky sometimes in their movements as they're doing this. As we've been studying Parkinson's, we know it's not the cause of it, but we know that Parkinson's disease causes a reduction of a special neurotransmitter in our brain called dopamine. Yes, they are aware of the fact that they're tremoring, but it's not something that they can consciously control. What they found is that people who have Parkinson's have very, very low levels of dopamine. They actually lose the ability to make dopamine. Yes, other conditions can cause it as well. Absolutely. Again, this is an elaborate neurological degenerative disorder. So again, I'm not going to be able to explain everything about it in, 10 seconds. But the point I'm trying to get at is I want you to understand how this relates to our blood-brain barrier. Many of the symptoms associated with Parkinson's are due to having low levels of dopamine, and they're not being, and their nervous tissue not being able to produce dopamine. What they have found is if you can get dopamine into these people's brains, many of their symptoms go away. But here's the problem. Dopamine cannot cross the blood-brain barrier. So can I give this person a dopamine pill that they can swallow or a dopamine shot in their arm and get it into their brain that way?
No.
Oh,
No.
No. So does anybody know how they used to get dopamine into the brain of Parkinson's patients? Nope.
like drink some alcohol before or something. I don't know.
That's not a bad guess. Yeah. Either injection into the spinal space, or they would actually drill a hole in the skull and inject it directly. All right. Now, not surprisingly, Parkinson's patients who even have very severe symptoms are somewhat hesitant to have holes drilled in their head so that they can get daily injections of dopamine. So it isn't a very reasonable way to resolve this. However, as I'm sure you guys know, chemicals, big complex chemicals like dopamine don't just appear out of nowhere. They're made. Building blocks are made, and you put those building blocks together, and there's this whole process that the nervous tissue does to do that. Well, what they found, one of the pieces that is needed to make dopamine is a chemical called L-Dopa. L-Dopa, the fancy term for it, is a precursor to dopamine. L-Dopa is basically one of the intermediate stages as the brain is making dopamine. And it turns out if you give the brain L-Dopa, the brain, these Parkinson's patients' brains can turn L-Dopa into dopamine. So their brains are able to convert this L-Dopa into dopamine. And the awesome thing about L-Dopa is L-Dopa can cross the blood-brain barrier. So you can take an L-Dopa injection or an L-Dopa pill, and it will get into the brain. And once it's in the brain makes dopamine. Parkinson's patient symptoms goes away. The stockholders of the pharmaceutical company are happy. Everybody's happy as a result of this. So again, by understanding how these things work, we are able to manipulate it to our advantages. All right. So there you go. So again, it gives you an example of how important that thing is. All right, let's talk about a second type of neuroglial cell. And again, we're still in the central nervous system. Our brain has surfaces, both internal, brain and spinal cord, because after all, that is our central nervous system, have surfaces. And those surfaces are both on the outside and internal. There are actually some fluid-filled spaces surrounding our brain and spinal cord. Anybody know the fluid that fills these spaces inside and outside of our brain and spinal cord?
The interstitial fluid?
It's not interstitial fluid, although that's a good guess. What is the fluid specifically that is bathing our nervous tissue?
By any chance epicranial?
No, that's no, although that can be a space. I know you guys know it. There it is. Although be careful, it's all one word. It's cerebrospinal. So it's not actually cerebral spinal. It's one word. Cerebrospinal fluid, right? Which, of course, as you guys may have heard of it, right? CSF, which you can abbreviate as when we are on an essay question or something like that. Cerebral spinal fluid bathes the surfaces, cushions the brain, holds it in place so it's not rattling against the walls as you move your head through space, provides oxygen, provides nutrients, provides chemical signals, tells the brain a lot about what's going on in the body. And so these spaces, both inside and outside, are lined with a special type of epithelial tissue, because after all, epithelial tissues line surfaces. Well, you might get partial credit for disc. I'll have to look at the question and see. But we have these epithelial cells, notice with these long microvilli to them. And these long, these long microvilli of these ependymal cells help to basically produce and regulate our cerebral spinal fluid. So they help to produce the cerebral spinal fluid and maintain the conditions of that cerebral spinal fluid. That plays an important role in both protecting and also, again, providing oxygen, providing nutrients, removing wastes, and providing chemical signals to the brain. So these ependymal cells line the surfaces and play an important role in making that cerebral spinal fluid. All right, questions on that? All right, excellent. Let us talk then about our third type of neuroglial cells. And again, as you can see, there's always going to be one or two of them. There's always that alphabet soup term that you know you're going to have to spell at some point or another. And here, this one in our neuroglial cells is an oligodendrocyte. Notice in some ways it is similar to an astrocyte, in that it has a centrally located cell body and it has some processes that come off of it. However, notice in this case, we have some axons, and I'll draw some examples of some axons here. All right, these axons, as we talked about, can be relatively long. And what these oligodendrocytes do is they take their plasma membranes and they wrap it around a piece of that axon, basically forming this huge layer of insulation. It basically wraps its processes hundreds of times around a small portion of an axon. Now, again, oligodendrocytes have somewhere between 5 and 20 processes, so it can wrap portions of multiple axons, in some cases up to 20 different pieces of them. And by wrapping around it, basically it forms this special insulation around the axon. After all, here in your house, you have wires in the wall. And if you were to cut open the wall and look in there, would you just see a bunch of naked copper wires hanging out there, bundled together inside of your wall?
No, they usually have some
hopefully
kind
not.
of plastic coating.
Yeah, exactly. They have some kind of insulation on it so that the signal coming out of it doesn't jump to another wire. So that when you turn on the light switch, your garage door downstairs doesn't open. And when you turn it off, right, you don't get your next door neighbor's, lights go on or something along those lines, right? We want to insulate it, and that insulation does two things. It isolates the signal, so it's not lost and doesn't move to another cell. But it also does one other very important thing. One of the super important things it does is it speeds up the action potential. After all, it is these axons that are producing the action potential, and this special insulation speeds up that action potential, makes the action potentials move faster. Of course, we have to have a special name for it. We can't just call it insulation, because we know our anatomists love to name everything. And this special name, as you can see right here, is a myelin sheath. That is the special name for this layer of hundreds of layers of the plasma membrane that it wraps around, or we say that it myelinates the axons. Plural, right? Up to 20. So in the central nervous system, these oligodendrocytes wrap around the axons, help to speed up the axons so that these axons can produce and quickly spread their axon from one part of the body to another. All right. Questions on that?
I have a question.
Yes.
So oligodendrocytes, is it the typical dendrocyte we see on a nerve, or this is a specialized?
So again, I think you guys get caught up in the vocabulary sometimes. You gotta remember the vocabulary of these words, they're all roots. Dendro just basically refers to something that sticks out from.
Okay.
So this particular cell has things that stick out of them. Dendrites are only found on a neuron. This is not a neuron.
Okay.
This cell does have processes that stick off of them. And since it has processes that stick off of them, that's why it's called an oligodendrocyte, right? Oligo, small number, right? A few dendrocytes, processes that come out and wrap around. So it has multiple processes that wrap around things, and that's where they get the name oligodendrocyte.
And
Small
where
number of things that stick out of it.
Where do we find these cells again?
Again, so far, all three of these types that we've talked about are all neuroglial cells of the central nervous system. So these are helping the neurons in the brain, helping the neurons in the spinal cord.
Okay, and just to clarify, so these hold on to the action potential, so it doesn't travel farther, but they
No,
also.
it doesn't hold on to the action potential. It wraps around the axon. Remember, the axon is specialized for producing the action potential. So that action potential spreads down the axon, like an electrical signal spreads down a wire in your house. And it's an electrical signal. It's like water. if you don't contain it, it can spread. It can, go other places, and we don't want that. So it acts like insulation to make sure that this action potential doesn't accidentally jump to some other neuron and do something we don't want it to do. So by insulating it, we isolate it to just this axon, and it also speeds it up so that it goes faster.
Okay, thank you.
Yep. So there you go. They form our myelin sheath around, and this one's got 1, 2, 3 different axons that it's wrapping around, forming a little bit of that myelin sheath. And again, protects it, insulates it, but probably the most important thing it does is it speeds up the action potential and makes the action potential go faster. And I think in our next class, we'll actually see how it speeds it up. If not the next class, then definitely next week. All right, that leaves us with one more glial cell. And again, we're still in the central nervous system. And this final one are what are known as microglial cells. Microglial cells are actually very similar to white blood cells in that they can move around the tissue and they look for dead or damaged tissues, and they can looking for things that don't belong, harmful pathogens that they can destroy. So when there is a trauma to the head, these will migrate to the parts of the brain that have been damaged to destroy any dead or damaged cells in that area, right? Congesting in that area, almost forming like a scar tissue in that area, and destroy any harmful pathogens that would get in there, which is really good and really important, except for what we've talked about before. Neurons are very specialized cells, and when that neuron dies, can we replace it with the same type? Do we regenerate nervous tissue?
Not really.
No. So instead, these microglial cells basically form scar tissue, right? They're really good at protection, but they form scar tissue. And as we know, scar tissue does not work the same as the tissue it replaces. So when there is a traumatic brain injury and that tissue becomes damaged and these microglial cells migrate to the area and destroy the dead cells, remove the dead cells, take care of any pathogens or harmful things that are in the area and all of that, they basically form a scar tissue. And now suddenly that part of the brain doesn't work the same way it did before. Traumatic brain injury. Again, it's a two-edged sword. It seems like a really, really good thing, but it also has the potential to cause damage as well. And no, typically we can't remove that scar tissue. Well, typically when someone's getting a brain surgery, it is typically to either remove something like a tumor that is growing in that area, or it could be if someone's having inappropriate firing of an area, it can be removed or something along those lines. The goal of the brain surgery is to limit the damage or minimize the damage that is formed. Now, it isn't completely hopeless. Because again, as we learn about the body, and in particular about the nervous tissue, the more we learn, the more we can study, the more we can interpret and change things. I'm going to tell you a story, and it is going to make you mad. So I will tell you right now 2 important things. One, this happened many, many years ago, right? And I promise you that everybody who was involved in this project died horrible, painful, slow deaths. So there's no reason to get your pitchforks and your torches and go running down to UC Davis or any place like that. because like I said, all the people there, I guarantee, died all horrible, horrible deaths as a result of this. All right, story time. At UC Davis, many, many years ago, they were very interested in studying, and they still are. They actually have an amazing mind institute where they're not doing these types of research there anymore. I'm telling you, they're not canceled. They're all dead. Many, many, many years ago, they were studying traumatic brain injuries, and they still do a lot of brain. They have the Mind Institute at UC Davis now, which is truly, truly an amazing program. But at the time, many, many, many years ago, they were studying rats. And rats are decent swimmers, but not great swimmers. So what they would do is they would take a rat and they would put a rat into a kid's pool that was pretty much filled with water. And the rat is able to swim his way around in the pool, and he would randomly swim his way around in the pool. And it turned out inside of the pool, they had put a clear plastic table under the water so it couldn't be seen. But what was able to happen is as the rat swam around, if he came in contact with it, he would be able to climb on top of it and stand on top of it, and he wouldn't have to swim anymore. So he would be happy. So they take him out, they take him back to his room, they give him his 3 square meals a day, and then they bring him back the next day. And the next day, they take him and they bring him and they put him in the water. And guess what happens when they put him in the water on the second day?
He swam.
He's looking for it.
Straight to the
Yeah,
plate.
and he's got a general idea of where it is, so he's able to find it much more quickly. And then the next day, he's able to find it much more quickly. And so basically, they time how long it takes him to find the table. All right, and he learns where the table is, and they time how long it takes him to swim there. Then they would take the rat. Let's not use rat. They would take the rat, and what they would do to that rat is they would connect a hollow tube right to the head of the rat with a standard length to it. They would then take a standard size weight and drop that standard size weight through that standard size tube to hit the rat on its head and induce a traumatic brain injury. They would then take the rat, and I told you, these all have died horrible, horrible deaths, so don't worry, it's okay. Horrible people died horrible deaths. They would then put the rat back into the pool, and what would happen?
It probably
You
would
would
start in the beginning, kind of swim all around.
Yeah, it took it much longer to find that table. Because, as we've talked about, what they did is they introduced a traumatic brain injury. And that traumatic brain injury lowered their function. But then they did the same thing to a second set of rats. And with the second set of rats, every single thing was done the exact same way with one big exception. Immediately after they induced that traumatic brain injury, the rats were given a special chemical, and that special chemical suppressed the function of the microglial cells. Normally, we talked about those microglial cells go to the area of the injury, aggressively destroy anything there that's bad, and form that scar tissue. But now they were given this chemical that stopped the microglial cells from doing that. And in those rats, when they put them into the bath after that, they found that they were much, much better at finding that table. This was a very important finding because what it told us is that while what these microglial cells do is good, they're very aggressive. when they get to a site of an injury, if a cell is dead, obviously it's going to gobble it up. If a cell is injured, well, the microglial cells, it's probably going to die anyway. I might as well just gobble it up. By being able to suppress these microglial cells, some of the cells that would have normally been destroyed by the microglial cells weren't fully dead. They were just injured or traumatized, and those were able to recover. And so as a result of that, by suppressing the microglial cells, we were able to better maintain function, brain function. We were able to reduce the amount of traumatic brain injury. And
So
this
is that something that they give to like humans now if they
absolutely. And this is one of many of these types of studies. Again, I think one of the places where you see this most prominently is in sports, because sports apparently are one of the most important things in the country. We have to have our sports, right? Let's placate the masses. And so if you think about even 10 years ago, if some football player had a spinal cord injury, he was pretty much in a wheelchair for the rest of his life, right? Whereas, gosh, 3 years ago now, 2 years ago, I guess it was 3 years ago, because everything's kind of fuzzy. 2 or 3 years ago, in Buffalo, on the very first game of the year, on the very first play, on the kickoff, one of their players on Buffalo damaged his had an injury, damaged his spinal cord, and he was actually back playing before the end of the year, right? Whether it's stabilizing the head, whether it's using medicines like this. We've also found that cold suppresses the function, suppresses inflammation, suppresses damage. A lot of what we use now to help to improve people with traumatic brain injuries, we have learned from studies like this. So while studies like this are indeed horrible to hear about, we have learned indeed a tremendous amount to help us to be able to maintain function and reduce the amount of traumatic brain injuries and help people who are in those that have these types of situations as a result of this. Right? So again, I'm not going to get into the sociological or the ethical debate about these types of things, whether they were good or bad. Many of these types of research projects can't be done anymore these days, or with a lot less, much more care, much more oversight and things along those lines. But it is very hard, while I will not justify what they've done, these types of experiments have provided us with a tremendous amount of information that has helped us to improve the lives of individuals who are getting into car accidents or falls or have other types of traumatic brain injuries as well. So again, there is knowledge is power. So I will leave you with that. Again, the ethics of this, I don't want to get into that discussion in that debate, but I do absolutely positively want you to know that this type of thing has tremendously improved the medical care that is available to us as humans. So I will say that. All right, excellent. All right, so again, just another example of how understanding how the body works is vitally important. All right, those are the 4 main glial cells that you need to know for the central nervous system. There are 2 that you are responsible for in the peripheral nervous system. And not surprisingly, they are going to be similar in the functions, because after all, neurons need a lot of the same stuff. So in our peripheral nervous system, we have 2 types with similar functions to those in the central nervous system. We see that first here with our satellite cells. These satellite cells, as you can see here, let's use green, get their name because they wrap around the cell body of this neuron. Notice they're wrapping around like satellites around the cell body. Well, again, it is a deep philosophical and sociological debate that we can get into, and it also depends on what you're studying. There's levels of everything. There have been, thousands of rabbits that have been tortured, for instance, just to make sure that we get the right shade of blush and that it doesn't wash off when you cry or so, you know, or exposed to water or something like that. So again, there's levels to all of these things. And like I said, we could have that debate, but again, that's a little beyond the scope of what we need to worry about for this right now. All right. Satellite cells, as their name indicates, because they wrap around cell bodies. Notice this cell body. What type of neuron am I looking at here structurally?
A
Unipolar.
Excellent. And based on that structure, what must its function be?
Sensory.
Excellent, right? So we have that. And of course, I will tell you now that this sensory unipolar neuron is found in a specific location. It is found in a ganglion of the spinal cord. Actually called, I'll give you the name. It's the dorsal root ganglion. So notice once again, we have this structure, function, and location. So here in the dorsal root ganglion, we will find a large number of these unipolar neurons. And the way you'll be able to tell they're unipolar is twofold. One is that it only has one process coming off of it. And the second is that it has all of these tiny little satellite cells wrapped around the cell body. These satellite cells basically are similar to the astrocytes in function. They do not form a blood-brain barrier. They're not forming the blood-brain barrier, but they are they do metabolize neurotransmitters. And they regulate ion concentrations. So they are similar to astrocytes in their functions that way. But notice also on, oops, let's do it this way. On the axon of our unipolar neuron here, there are these small blue cells that have wrapped themselves around it hundreds of times, forming our myelin sheath. However, what's happening here with these particular cells is this, here, I'll change colors. This right here is a single cell. One cell wraps around one small portion of an axon hundreds of times. And these one cells doing this one portion are what are known as Schwann cells. Here we see a slightly better illustration of how this works. Notice here we see an example. So again, each one of these is a Schwann cell in its entirety. Notice here is the cell. It takes its plasma membrane and it wraps itself hundreds of times around that axon, forming that myelin sheath. This type of an axon that is myelinated, we call a myelinated axon. And like we said, that myelin speeds it up and insulates it. Now, not all neurons, let me rephrase that. Not all axons are myelinated, but they still need to be separated. And it turns out our Schwann cells do that as well. Notice that's what we're seeing going on over here on the right side of the screen. Notice again, here we have a single Schwann cell. And notice what it's done is it's taken 9 different axons and it's keeping them away from each other. But notice it's not wrapping around any of them 100 times. It's not forming the myelin. So these are actually unmyelinated axons. So we still need to keep them apart because we don't want our wires to cross. Just like in our wall, we don't want our wires to cross. But They're not wrapping around it 100 times. So these are typically unmyelinated axons. So our Schwann cells protect the unmyelinated axons, and they form the myelin on our myelinated axons. All right. Again, this myelin sheath, multiple layers of the plasma membrane, all those phospholipids and proteins wrapped around hundreds of times. What are the two cells that produce our myelin sheath again?
The Schwann cells and oligodendrocytes.
Excellent. And where do the Schwann cells produce the myelin?
On the axon.
Where? Central nervous system or peripheral nervous system?
Or peripheral nervous system.
Excellent. And oligodendrocytes in the central nervous system. Excellent. Perfect. And again, here we see that example wrapping itself hundreds of times. Now, I want you to notice one of the things that hopefully you already noticed, but I want to show it to you here in this picture. Notice whether it's an oligodendrocyte or whether it is a Schwann cell. Notice when they're forming that myelin, it's not continuous. There is always a little gap in between. That gap in between is simply referred to as the node. However, as you can see, good old Bob Ranvier was the first person who identified these little gaps. So, of course, they're named nodes of Ranvier. And Ranvier is incredibly fun to say. So you're welcome to call them the nodes of Ranvier, but you can also just call them node as well. All right. And again, it insulates and protects, but most importantly, it speeds up the action potential, which is going to be super duper important for us. All right, we're on time. Good, excellent. Questions on this?
Yes.
Okay, go ahead.
Do oligodendrocytes also speed up the thing?
Yes, both the oligodendrocytes and the Schwann cells make myelin sheath, and myelin sheath speeds up the action potentials.
Okay, but oligodendrocytes don't have the node. Would that affect how fast it goes?
So great question. So here's the easiest way to do this is let's look at the example here of 3 neurons, 3 axons. Here's 3 axons in the peripheral nervous system. And on this, every single piece of myelin, this piece of myelin and this one, and this one, and this one, and this one, and this one. I'm not going to bother myelinating the whole thing, but you get the idea. Every single one of these blue things I'm drawing is an individual Schwann cell. So every single one of these are individual Schwann cells forming one piece of myelin on one axon. Okay? The difference between the Schwann cells and the oligodendrocytes is here are 3 axons in the central nervous system. A single oligodendrocyte will have multiple processes. So it could form a piece of myelin right here, and also a piece of myelin right here, but also a piece of myelin right here, and also a piece of myelin right here. Whereas a second oligodendrocyte with its processes could form the piece of myelin that sits here and the piece of myelin that sits here, and 2 pieces on this one side by side, and maybe an additional one down here. Notice one oligodendrocyte can make a piece of myelin on multiple axons or in multiple locations on a single axon. So one cell in the oligodendrocyte can form many pieces of myelin, whereas a Schwann cell can only produce one piece on one axon. But that's just how they make the myelin. Once the myelin is on the axon, whether it's a Schwann cell or an oligodendrocyte, it works the same way. And so, yes, there's going to be nodes in both. Does that help to make sense of it?
Yeah.
Okay, excellent.
I have a question too.
Yep.
So from my memory, we were supposed to have 3 of each kinds of cells from the central nervous system and the peripheral.
4 in the central nervous system, 2 in the peripheral.
Oh, okay. Okay. Then I got my notes are correct.
Yep. Which again, if you think about it, makes sense. 90% of our neurons are in the brain and the spinal cords. And it's not surprising that there are far more neuroglial cells in the central nervous system than there are in the peripheral nervous system. All right. Questions on that? All right, excellent. We've got time for one more break because now that we know the anatomy of these things, we can start talking about the functions. Some of this is going to be, let me say this again, most of this is stuff that we've kind of already talked about and hit on with other parts when we were talking about muscle cells or when we've talked about neurons in other ways and things along those lines. So some of this is just going to be adding vocabulary, but some of this will also be adding a little more depth and understanding to the things that we've talked about. So we need to talk about a little bit about the mechanics by how we produce electrical signals in a neuron, these potentials that we've been talking about. And again, we've been talking about action potentials, but it turns out there's actually 2 types of potentials: action potentials and graded potentials. So we need to talk about both of those and how they're different and how they're similar and how we make them and all of that type of fun stuff. So we have some function to talk about because what we will do, today's Thursday, right? What we will do on Tuesday when we come back, we are going to learn about the almighty action potential. Everything you ever wanted to know about the action potential and more. We are going to do it. It is one of the more, I mean, it's not the longest. Obviously, the muscle contraction was the longest, but it is definitely one of our bigger physiological processes for this section. So we're going to do that. But we need to lay some groundwork first before we can dive into that action potential. So we're going to talk some vocabulary. We're going to talk some explanations, make sure we understand all these basic building blocks so that we can then see how our action potential is going to work. So let's go ahead and take one last break. We'll take a 15-minute break. So it's 3:22 right now. So let's just call it, we'll come back at 3. Let's do 3:35 so that we have enough time. That gives us an hour. A little bit of a shorter break, but that's okay. 3:35, and I will start the recording at that point. I won't use the whole hour, but I want to have more, make sure I have enough time to make sure that we understand these concepts. So I will meet you back here in 12 minutes. All right, so see you guys shortly. All right. So we've started talking about some anatomy, and now we can start talking about how these neurons are going to function. As we talked about, neurons have a characteristic where they are irritable. And again, irritable means that they basically receive a stimulus and respond to it. We said that they're electrically excitable because they respond by producing an electrical signal. And that electrical signal is what we call a potential, like the action potential. But it turns out that there are 2 different types of potentials. The first is an action potential. Remember, this one not only is big, but we know that this is a big positive signal. Does anybody actually remember how big of a positive signal we actually said it was? I think we mentioned it briefly when we were talking about the muscles. Does anyone remember how big it gets?
What is it, negative 70 millivolts?
It starts at negative 70, and remember we had to get the threshold to get it. But then,
Was
does
it
anyone remember how
55?
big?
Wasn't it 50, at least 55?
100.
There we go. There is a positive 100 millivolt change. It is a big, huge positive signal. And again, that's really not an average. It is fairly stable. It is fairly uniform. And as we see here, it is what we also call all or nothing. The best example I can give you for this is a light switch. I have a light switch on my wall. And if I push it up, the light goes on, right? Now, if I only push it halfway up, does the light only halfway come on?
No.
No. If I push it up twice as hard, does the light come on twice as bright?
No.
No. This light in this room is basically like an action potential. It is a big, bright signal that is all or nothing. It is either on or it is off. And remember, the other big important thing we know about an action potential, it can only occur along the axons. It can't occur on the cell body. It can't occur on the dendrites. They cannot produce action potentials. But what they can produce is a signal known as a graded potential. These are small variable signals. And I do mean small. On average, these are around 2 millivolts. Very, very different from 100. And they're variable. If we want to stick with the light switch analogy, while here in my office, it's on or off, in my dining room downstairs, I have a different type of switch. It's a knob where I can adjust the intensity of the light, right? If it's just me and my wife having dinner together, we like to turn the light up nice and bright so we can see each other and communicate and enjoy each other's company. However, if the children are with us, especially when they were younger, you turn that light down nice and low. You don't want to see all the mess they're making all over the table and all over their faces and all over their floors. You don't want to make eye contact with them. And that's a lot easier to do when it's dimmer in there. So we're able to vary the amplitude. It can be slightly bigger or smaller. And yes, question.
Yeah, I have a question. So action potential is our brain telling our body what to do, but is there a reverse where something that we feel tells our brain what to do?
Action potential is not your brain telling your body what to do. An action potential is just the big electrical signal that is sent so that we can send information from one part of our body to another. What
So it's
that
gonna be the
information
same.
does and how it is interpreted is much more complicated than that. So all we're doing is producing electrical signals. It's very much like water, or it's very much like the wiring in your house, right? When you turn a light switch on or you switch a light on the wall, an electron moves along a wire, right? What that electron moving along the wire does totally depends on what it's connected to. If it's connected to a light, it turns the light on. If it's connected to the garbage disposal, it turns on the garbage disposal. If it's connected to the garage door, then the garage door goes up, right? All we're doing is producing that signal, producing that current that is spreading along the wire. Once it gets to the end, what it does is something totally different.
Okay, so the input versus output, it's still the same thing.
Well, it depends on what you mean by input or output. If you think of, so let's do this. We have a neuron that wants to communicate with whatever the target is. It doesn't matter what the target is. There's some cell here that is the target. as we have just finished talking about, and as we've mentioned, we know that on the axon, we're able to produce a big, huge positive signal that can spread all of that distance down to the end. Of course, remember, as we talked about, we have to make that action potential at the trigger zone in the axon hillock. And the question becomes, how do we make that? Right? Because after all, our cell body and our dendrites, our cell body and our dendrites don't make action potentials. So how the heck do we make an action potential? Well, it turns out the way we make it is out here on the cell body and the dendrites, we have graded potentials. Here we can produce a small signal and spread that small signal to the axon hillock. Or we can make 3 small signals and spread it to the axon hillock. And if at the trigger zone, those graded potentials are big enough, then we get the action potential and it spreads down the axon. So if you're thinking in terms of input or output of the neuron, the graded potentials are the input coming in, the action potential would be the output going out. But again, at this point, we're just talking about producing this electrical signal and electrical signals, plural. Actually, you know what? Let's go ahead and leave that. I like that. Just want to get rid of the blue one. There we go. Oops. There you go. Perfect. So action potentials occur along the axon. Graded potentials occur on the cell body and the dendrites. They're small, they're variable. So again, like I said, they can be bigger or smaller. And if you notice, they can actually be positive or negative. Whereas that action potential is always 100 millivolts positive, graded potentials can actually be positive or negative. All right, so these are the electrical signals we're going to be producing on our neuron, and we have to figure out how. How are we able to produce that? Well, remember, when we're talking about a wire in your house, and we are talking about that electron is a charged particle. And anybody happen to know the charge of an electron?
-1.
Yeah, well, it's negative. That negative is all I was looking for. It is a charged particle, and that charged particle moves. Nope. That charged particle moves down the wire. And when that does that, basically, it produces a current. All right? That's our electrical signal. It's producing a current. Well, that's what we need to do here. We need to move charged particles. And while our cells don't necessarily have the electrons in them that move, they do have things like sodium and potassium and chloride, right? Those are charged particles, cations and anions. And if we can get them to move into or out of a cell, then basically we're moving a charged particle. We can produce a current. And so that's what we need. We need a reason for them to want to move and the ability for them to move. And if we can do that, we can produce a current, either an action potential or a graded potential in our cell. Now, a neuron, like most of the cells in the body we've talked about, at rest have a resting membrane potential. They're polarized. There's a different charge inside and outside. And what did we say the resting membrane potential of a cell in the body is? Yeah, -70 millivolts. All right, so actually let's do it this way. Let's go to our whiteboard. Here is our cell. And as we know, we have a resting membrane potential. That you guys have just told me is negative 70 millivolts. Excellent. So basically, there is an imbalance in charge. Inside and outside. Let's make that a little smaller. Of the cell. And if you remember way back when we were talking about cells, we called this an electrical force. But remember also we talked about how there is an imbalance of ions inside and outside of the cell. Like, for instance, sodium. Where is there more sodium, inside the cell or outside the cell?
Inside the cell.
Outside.
Yeah, excellent. So for sodium, remember there is a lot more sodium outside the cell. What about potassium? Where was there more potassium?
Inside.
Inside the cell. Where was there more calcium?
Outside.
Outside the cell. Where was there more chloride?
Outside.
Outside. Right? So we have this imbalance of ions as well. And remember, we called that our chemical force. So our electrical force and our chemical force influence the movement of our ions. So if you remember correctly, as we talked about, sodium is a positive ion. The inside of the cell is negative, so it wants to come in down its electrical force. There's more sodium outside, less sodium inside, so it wants to go down its chemical force. So remember, for instance, we talked about sodium really wants to get into a cell. All right? So any of that sound vaguely familiar? We've talked about that before.
Yeah.
Okay, excellent. So these ions basically want to move. We could say they have a driving force. So if a channel is open, ions will move to reach their equilibrium. And we'll identify that and talk about that a little bit more in a minute. Okay, again, not really super new information. This isn't terribly new either, but as it turns out, there are two types of channels in a cell. The first of these types of channels, let's actually do this way, are what are known as leak channels. And with a name like leak channels, guess what they do?
Like slowly control.
Well, can you really control a leak?
Or well,
No.
like lets things in and out,
Yeah.
like in very small amounts, I guess.
Basically, a leak channel is really just a hole in the cell. Basically, leak channels are channels that are open all the time. And in fact, and let's make these blue. Our cell has tons of leak channels. One of the leak channels that it has are sodium leak channels. So if I have a channel that's open all the time for sodium, guess what sodium's doing all of the time?
Leaking in.
So
Yeah, it's sneaking into the cell, absolutely. And guess what? We also have, oops, didn't want that. Potassium leak channels. So guess what's always happening there?
Leaking out.
leaving the cell.
Yeah, potassium is constantly leaving the cell. That could be a huge issue. We constantly have sodium sneaking its way in. We constantly have potassium leaving. And that could really affect the ability of this cell to do work. Luckily, this cell happens to have a particular type of pump on it that is going to allow it to compensate for it. Anybody remember what the pump that's going to help us with this while the cell's sitting at
Calcium
rest is
pump?
going to be? Say again?
Calcium pumps?
One more
Sodium,
time.
potassium.
The calcium pumps?
Not the calcium pumps, although the calcium pump will help us with that calcium that leaks in. No, there's something that's going to help us with both sodium and potassium.
Sodium potassium pump.
There you go. Remember we talked about those sodium potassium pumps, or again, what we call the sodium potassium ATPase, right? Both of those were acceptable words for that. And someone again, remind me what this does. This uses ATP. So let's put, oops, I don't want, let's leave this green. But what I do want to do is,
Are they the ones that pump 3 potassiums to 2 sodiums?
close. You've got the right idea. This is going to use ATP, and it is going to use ATP to kick sodium out. So that sodium that comes in gets kicked out. It is going to bring potassium in. So as that sodium comes in, it gets moved right back out. As that potassium leaves, it gets brought right back in. But somebody remind me of the numbers. How many sodium does it kick out?
2.
2.
2?
3?
3. 3 sodium out. And how many potassium does it bring in?
2.
2.
2. There you go. So 3 sodium out and 2 potassium in. There you go. And that helps to maintain those balances. So notice, this is still a cell at rest. When a cell is at rest, it's using ATP. Ions are constantly coming in and out, right? But there's no net change. Remember, when it's at rest, there's no net change, and we can't modify this because these channels can't be opened or closed. So it's doing this all the time. There's no net change, and we can't modify it. So it really can't do work for us because we're not able to change it. So if all we had were leaked channels, we wouldn't be able to get this cell to do any work for us. But lucky for us, as you know, we have a second type of channel. And I've already used all these other colors, so let's just go with black. The second type of channels, as you know, are gated channels. And gated channels, as the name indicates, can be open or closed. Right? Whoops. They can change states. So, as we've seen in the past, if I have a calcium channel that is gated, that sometimes can be open and sometimes can be closed. Sometimes I can let calcium in, and sometimes I can stop calcium to go in. And by being able to change it, sometimes come in, sometimes not, I can change the condition of the cell and I can get the cell to do work. That's the real key to these gated channels. With these gated channels, I can change the flow of ions that changes the cell, and the cell does work. All right, we open up that voltage gated calcium channel. Calcium came in and the neuron releases its acetylcholine. And I've already partially given it away, but you guys actually know this. There are actually 3 specific types of gated channels. What are the 3 specific types? What are the 3 keys that can be used to unlock these gates?
Chemically.
Voltage and chemical.
Excellent. Chemically gated, we need some type of chemical binds to it and opens it. Second was, I heard you guys say voltage gated. How do we get voltage gated channels to open?
Action potential?
No, the action potential is what we get when we open the voltage gated channels. How do we get voltage gated channels? Well, okay, you're right.
Depolarization.
The voltage gated came down, but what did that action potential do that caused the voltage gate to open?
Electrical stimuli?
Okay, so it changed the membrane potential.
Depolarize it.
Excellent. Depolarized it to what magical point?
Threshold, -60.
There you go. To threshold. And excellent. Since you said it, we'll go ahead and put that in there. -60 millivolts. Excellent. So for a voltage gated, we have to change the membrane potential to reach threshold. That's the magic term, which happens to be -60 millivolts. Does anybody remember what the 3rd type of gated channel is? I know we've actually talked about it, but it has been a very long time.
The ligand?
Concentration rated.
No, remember, ligand just means chemically gated. Ligand is just a fancy word for chemical.
Concentration.
Is it
Not a
ATP?
bad guess.
ATP, ACE, or something like that?
No, because then those are pumps. You guys have good ideas. Now, I wasn't sure. I wasn't sure we talked about it in this way. But if you remember way back when we were talking about the skin, we were talking about those Pacinian corpuscles, those onion-shaped sensory structures that we talked about how when you squeezed it, it stretched the membranes. And when you stretch the membranes, that opened the channels and we perceive pressure. So the 3rd type of gate is what we call a mechanically gated. For a mechanically gated channel, we change the shape of the membrane. And when we do that, we let the ions flow. Now, I've done this with all the words, but we've got some actually very great pictures that do a nice job of pointing this out. So let's come back here, right? Again, we've got this unequal distribution of ions. We have our sodium, potassium, ATPase, which helps to keep kicking things out that sneak in those leak channels. And here's the pretty picture from the textbook that shows that. Again, that pesky potassium keeps leaving, and so we use ATPase to bring it back in. Sodium keeps sneaking in, and we use that ATP to kick it back out. And 3 sodium and 2 potassium coming in, and that helps us stay at that -70 millivolts. Right? And again, we talked about that chemical gradient and that electrical gradient. Positive moves to negative, positive moves away from positive, and things move from a high concentration to a low concentration. We did that when we talked about cells, right? But that makes the ions want to come in or want to leave, but to leave, they have to have channels. Our leak channels are the non-gated channels that are always open, and our gated channels, voltage gated, chemically gated, and mechanically gated. Now, here's what I wanted to show you. We've got these great pictures. And by great pictures, I mean very simple illustrations. but it does a nice job of showing an important point. Here is our chemically gated channel. And notice our chemically gated channel has two states. If there is no chemical bound to it, it is closed. And if the chemical is bound to it, it is open. So notice here, our friend acetylcholine binds the channel and opens it. No acetylcholine, the channel is closed. Very simple, very straightforward. Here is our voltage gated channel. Notice with our voltage gated channel, it has what we call, that's what I want, right here, our activation gate. When that activation gate is closed, the channel is closed. And of course, when we get it to open, then the channel is open. And how do we get it to open? As you guys have mentioned, we get it to reach threshold. When it reaches threshold, and that threshold happens to be that magical point of negative 60 millivolts, that's when that door swings open. Now, this is a very, very simplistic illustration. Your textbook's got a great picture that I'll show you in a minute of how this works. But I want you to notice that some voltage gated channels have a second gate called the inactivation gate. If we were to look at this on a real channel, what you would notice about it, here, I'll just draw one real quickly right here. There's our channel in the plasma membrane. This inactivation gate is made up of two proteins. One is a long linear protein, and one is a big globular protein. I make a point of mentioning this because it has a very technical name, which you can only appreciate when you look at it. And the technical name for this inactivation gate is the ball and chain. Now, what can happen with this ball and chain? So notice our activation gate is open. It's out of the way. But what can happen is that the ball of our ball and chain can actually come up and wedge itself into the channel. If it wedges itself in the channel, can anything come in anymore?
No.
No. Notice if we put that ball of the ball and chain here right now, our activation gate is open, but still nothing can flow through it. And so in this case, this type of channel is said to actually have a third state. It can be open, it can be closed, but if that ball and chain wedges itself in there, then this channel is said to be inactive. And here's the problem with it being inactive. When it's inactive, it cannot open again. At least not right away. Cannot open right away. Oops. It must close first. So what happens is when the channel becomes inactive, it has to first close, and then once it closes, then it can open again. It is not possible for it to go from inactive to close. And we'll see why that is when we look at the picture from a textbook that actually shows what this channel really looks like. So give me a second, and I promise to make a little bit more sense of that. Notice here is our last channel, that mechanically gated channel. Notice it also has two states, closed and open. And notice when you push on the channel, push on the plasma membrane, it opens the channel. When you stop pushing on the plasma membrane, it closes it again. So chemically gated, voltage gated, mechanically gated. We have talked about all of these before. So we didn't necessarily use the term mechanically gated when we were talking about those, but we did talk about how when you stretch the plasma membrane, it opens. And we haven't talked about this inactivation. Those are really the only two things that are new that we've talked about here. Now, what I want to show you is that great picture from your textbook. Notice here we have a great picture from your textbook of an example of a voltage-gated sodium channel. And now you really see how this works. The way this actually works, notice this is our two proteins that form the activation gate. And it turns out they're very positively charged. So when the cell inside depolarizes to threshold, these negative gates don't want to be here where it's positive, pardon me, these are positively charged. When the cell depolarizes, these positive doors don't want to be inside anymore. And so what happens is they swing up out of the way. And when they swing up out of the way, our channel is open. And now it can flow. But then that pesky ball of the ball and chain wedges itself in here. And now the ions can't flow anymore. Now our gate is inactive and it's stuck there. But notice when we go back below threshold, those activation gates swing back down. And when they swing back down, notice they pop the ball out. And now it's not inactive anymore. And now it can go to open. So basically what happens to these channels is they go from closed to open, open to inactive, and then they have to go all the way back and close again before we can open them a second time. All right.
So the positive charge causes those two proteins to change their shape, but what causes that globular protein to enter the channel?
That is an awesome question. And that was a question that was asked for many, many years. And then they were finally able to figure it out. What happens is that ball, like all of the components inside of our cell, they have kinetic energy and it is just randomly moving around. And as it randomly moves around, it's going to wedge itself in there. They actually figured this out because what they were actually able to do, again, the field of neuroscience, the scope of it is amazing. There are people who spend their entire lives just looking at the amino acids that are making this entirely one channel. And what they were able to do is they were actually able to manipulate these channels where they could change the length of the chain. They could make the chain shorter and they could make the chain longer. And what they found is when the chain was shorter and there was less place for the ball to move, it would wedge in much more quickly. Whereas when they made the chain longer, it was much more random in where it could move, and it took much longer for it to just randomly wedge itself into that, into the pore. So it's really just the random movement of it. There's no driving force on it. It's just randomly bouncing around. And when those activation gates are out of the way, it can sometimes wedge itself into that channel.
Okay, thank you.
Yep. All right. Now, the last thing, how are we doing on time? Excellent. The last thing we need to talk about from today is, as I mentioned, our ions are 2-year-olds. What do 2-year-olds care about?
Surviving.
True, surviving, yes. So when you ask them, they want to make sure they have a very important, 401k, right? That and a roof over their head and all those kind of things. Yeah, they care about survival, but what do they really care about?
That everything goes their way.
Yeah, they only care about themselves. All ions care about is themselves. They only care about themselves, right? If you have a cookie that a 2-year-old wants, they will cut you to get it. right? They only care about themselves. Sodium only cares about itself. Potassium only cares about itself, right? Calcium only cares about themselves. They don't care about the cell. They don't care about the other ions. All they do is care about themselves. And what ions want, there you go, exactly. What ions want to be at equilibrium. Now, remember, equilibrium for an ion is a little bit tricky, right? If we were just dealing with glucose, as we've talked about before, and I have my beaker filled with water, and it's 65% glucose on this side. Doesn't need to be capitalized anymore. And it is 25% glucose on this side. And we poke the holes in the membrane. Poke, poke, poke. What's going to happen? There you go. What's going to happen if we poke holes in our membrane here with our glucose?
It's gonna reach equilibrium.
And what does that mean? You are 100% correct, but describe to me how it reaches equilibrium and what equilibrium is.
When the movement from right to left is equal, like they keep moving.
It's going to move down its concentration gradient until what happens?
The concentration is equal.
All right, and what would that be in this case?
It would be 45.
Yeah, 45% on this side.
Or 40.
45% on this side. Nope, I think it's 45. 45% on both sides, right?
Yeah, sorry.
Yeah, 45% on that side. Now, at that point, is there no more movement of glucose? Now, remember equilibrium.
No, there's still movement.
Yeah, there's still movement, but there's no net change. That would be equilibrium, right? So we did that. We remember that from before. The problem when you do something like that with sodium, right? If there was 65 sodium on this side and there was 25 sodium on this side, equilibrium wouldn't be 45 sodiums on both sides. Because remember, when we're dealing with a charged particle, every time one sodium moved to the right, over here would get a little bit positive, and over here would get a little bit negative. And remember, sodium likes negative things. So while we don't need to worry about the math for this, when we're dealing with ions, equilibrium is when the chemical force and the electrical force are equal and opposite. We don't have to worry about figuring out the math of what that means, and we don't even necessarily need to understand what that means. All we need to know are the numbers involved in this. And those numbers involved in this, I want to leave that here, so I'll just go through and remove all of this stuff. Basically, as we know, the cell is happy when it is at negative 70 millivolts. But sodium doesn't care about the cell. Sodium wants to be, so the equilibrium potential of sodium is positive 66 millivolts. So basically what sodium wants to do is move to reach equilibrium. So notice if you open a sodium channel, Sodium enters the cell to make it more positive, to try to reach positive 66 millivolts. So sodium is going to keep rushing into the cell, making the inside of the cell more and more and more and more positive till it reaches its equilibrium. And if it's able to reach its equilibrium, sodium would happily stay there forever. Potassium, as we know, likes to leave the cell because potassium wants to make the cell more negative. And in fact, potassium equilibrium potential of potassium is equal to negative 90 millivolts. So notice when we open a sodium channel, pardon me, a potassium channel, potassium leaves the cell to make it more negative, to try to reach negative 90 millivolts. So the take-home message of all of this is this right here. Ions want to be at their own equilibrium, and they will move to make that happen. So as we've already we know when you open a sodium channel, sodium rushes in. Now we know why. It wants to make the cell more positive. It wants to reach positive 66 millivolts. The cell doesn't want it to reach positive 66 millivolts. The cell's not going to be happy there. Potassium definitely doesn't want to be at positive 66 millivolts, but sodium just cares about it. So if you open a sodium channel, it's going to try to reach positive 66 millivolts. And if you open a potassium channel, potassium is going to leave the cell to make the cell more negative because it's going to try to reach its equilibrium potential. Basically, the equilibrium potential is where the ion will be happy, where that ion will be at equilibrium. And that's its goal. Its goal is to reach that. If you haven't figured out at this point in this class, I'm not a huge numbers guy. Numbers are usually the kind of things that you can look up. But every once in a while, we run into numbers that are important. These numbers are resting membrane potential, our threshold. Those four numbers are numbers you absolutely positively need to know. And since I just mentioned it, what is our threshold value again?
Negative 60.
Excellent. So we need to know our resting membrane potential. We need to know our threshold. We need to know the equilibrium potential of sodium and the equilibrium potential of potassium. These four numbers you definitely need to memorize, because these four numbers we need to know to understand how the neuron does its work. And again, let's put it all together and bring it together. When you open a sodium channel, sodium comes into the cell. A positive ion crosses the membrane. When you open a potassium channel, a positive ion leaves the cell. A positive ion crosses the membrane. Those charged particles moving either into or out of the cell produce a current, right? Produce that electrical flow. And of course, that electrical movement we call a potential. So graded potentials, action potentials are the movement of charged particles, ions, into or out of the cell. All right, this is the information you need both for lecture on Tuesday, but also this is going to help you to understand what's happening in your action potential labster. You're going to do it on the labster, and like I said, hopefully it'll make sense, hopefully it'll work, but if not, we absolutely positively will be going over it together in class on Tuesday. We will talk about how you not only make an action potential, but how we're going to spread that action potential as well. We're going to do all of that next week. All right. Questions on that? No. Graded potentials are what are produced on the dendrites and the cell body of a neuron. Action potential is what is sent on the axon of a neuron. All right. So whether it is in the brain, whether it's in the spinal cord, whether it's in the arm, it's about the neuron. It's not about the big structures. We're just talking about the neuron. Now, that being said, we are going to talk about the big structures. Let me do that with the last 2 minutes that I have left. We're not going to do anything with this yet, but I want you to get started and I want you to be prepared. Where is it? Is this it? Yeah, this is what I want. All right. As I mentioned before, I'm sure since Tuesday, you have been poring over the modules that you are responsible for this section. On the module, you will find a handout on the brain anatomy, the central nervous system anatomy. That central nervous system anatomy and the histology that goes along with it are probably going to be about 50% of your next lab exam. So it is a huge part of this class. We will spend some time going over it, but you need to start looking at it ahead of time. Now, notice we had 206 bones. We have a ton of muscles we had to learn. And one of the things we did to make it easier to identify them is break it down into smaller, more digestible pieces. Instead of saying, here are all the muscles you need to know, first, we studied the muscles of the head, and then the muscles of the neck, and then the muscles of the trunk, right? For the bones, we did the skull, right? The axial skeleton, starting with the skull, then the vertebral column, then the bony thorax, and so on and so forth. We broke it up into bits meaningfully. And we can do the same thing with our brain as well. What's cool about this handout that you see, or no, this illustration, I should say, that you see right here, is it shows the very early development of the brain. The way your nervous tissue forms is it actually form a flat layer of cells. These flat layer of cells then get a crease in them that causes them to fold up, and it rolls up into a tube. And that's what you see right here. You see an example of, at about 3 weeks of development, there is this tube of nervous tissue that forms. And if you look here, by week 6, this tube has divided up into 1, 2, 3, 4, 5 different enlargements. These enlargements are what are referred to as the cephalons. These cephalons, as you can see, are the telencephalon, diencephalon, mesencephalon, metencephalon, myencephalon. And I appreciate that there are 3 M's there, and that can be a little bit confusing. But I will also point out to you that they're in alphabetical order. Mes, and then met, and then my. These 5 enlargements correspond to 5 distinct regions in the mature brain. The telencephalon becomes a big, large structure we call the cerebrum. The diencephalon actually stays a region called the diencephalon. The mesencephalon becomes a region called the mesencephalon. The metencephalon becomes structures known as the cerebellum and the pons. And the myencephalon becomes everyone's favorite, the medulla oblongata. And notice if you look at the colors of these here, they correspond to the colors here in the mature brain. So here in a mature brain, we see the cerebrum, which is the telencephalon. This oval-shaped region in the middle of the brain is the diencephalon. The little green is the mesencephalon. This two big blue enlargements are the metencephalon, and there's the medulla oblongata, the myencephalon. So if you notice on your list, all of the brain structures you're responsible for, we divide up into these cephalons. So first we'll learn the things of the telencephalon, then the diencephalon, then the mes, the met, and the myencephalon. So we'll go through them in order. And it's just, again, the same way that we can break the skeleton into the skull and the arm and the foot, we can break the nervous tissue into these cephalons as well. So again, they're on the list and I don't want you to be confused by it. I want you to understand where they come from. And so, like I said, you see this nice color coordination that kind of shows these regions and what they are. And these are all straight from your textbook and resources like that. So start looking at that. But like I said, we will go over that together. And like I said, these structures, the parts of the brain here and what they do, and the histology with this is going to be about 50% of your lab exam. So this is a huge, huge part. All right, questions on that? All right, excellent. One day in, but we actually got as far as I'd hoped for. So we're actually kind of on pace. So that's a shock and a happy thing to have. It won't stay that way. Yes, question.
Do you have any tips for how we can start working ahead on memorizing stuff we will need to know for the exam?
So for the lecture, obviously your lecture outlines tell you exactly the things that I'm going to be talking about. So you can look at the lecture notes for the outlines. You can look at the part of the textbook. So for instance, I've already told you, we're going to talk about graded potentials and action potentials. So you can look at the notes from the lecture outlines for that, and then go to the textbook to look at those things to help you that way. For the anatomy, you've got your study guides. So for the histology, look at your resources that are on the Master in A&P, look at the websites I've given you, look at your histology atlas, and your textbook is pretty good for the anatomy of the brain. So for the regions and the functions and their structures is a pretty good resource for that as well. So I would focus on the tools that I've given you. Basically, the lecture notes are the study guide for the lecture exam. The lab handouts are the study guide for the lab exam. So use those as your focal points for what it is that you're going to study. All right. All right. Yes. And of course, again, remember, as always, I say that when I'm going over this stuff with you, the goal when I'm done talking isn't for you to have mastered this information. The goal is that you've been exposed to it, have some experience to it, so that when you're studying on your own, hopefully it makes sense. You can recall the things we've talked about, and it can help you to make sense of this dense, what can sometimes be dense material. All right. Excellent points and excellent questions. Any others? All right, then you guys have a happy and safe weekend. Again, I believe everybody in this class is now over 16. So remember, today is the 15th, meaning that everybody is eligible now for the vaccine. Depending on if you're in Sac County or Placer County or those types of locations, please look at their websites. There are great resources where you can go and you can get your vaccinations. I strongly encourage you to take advantage of that. All right, excellent. You guys have a great weekend. Yes, absolutely. I encourage you strongly to do that. I have done it. My wife has done it. My daughter, my oldest, my youngest isn't young enough, but my older one is 16, and she got her first shot already. We had to go to Davis to get it done, but we were able to get it done through UC Davis because they had gotten ahead earlier this week. So she was actually able to jump the gun on the 15 because they were ahead. So yes, I strongly encourage you guys to take advantage and get vaccinated. Be safe, be smart. All right, have a good weekend, and I will see you guys on Tuesday. Bye.